Hello there and welcome to Biology with Charles, where curiosity slows down just enough for us to examine the small details that quietly shape our lives. Tonight, we're turning our attention to something you've felt many times, even if you've never seen it, measured it, or consciously noticed it working behind the scenes. It's involved in the warmth of a hug, the comfort of holding someone's hand, the calm that settles in when you feel safe with another person. It plays a role in romance, in trust, in friendship, and even in the bond between a parent and a newborn child. We're talking about oxytocin, often nicknamed the love hormone. That name sounds simple, almost tidy, as if one small chemical neatly explains one of the most complex experiences humans can have. But biology is rarely tidy. Oxytocin does not float around your bloodstream with a tiny arrow pointing toward romance. It doesn't guarantee loyalty or devotion. It doesn't write poetry or plan anniversaries. What it does is influence systems that shape how we connect, attach, and respond to one another. Before we go any further, I'm curious. Where are you listening from tonight? Are you at home, winding down after a long day? Is it early morning or well past midnight? And when you think about the people you feel closest to, what's the first memory that comes to mind? A shared laugh, a quiet conversation, a simple moment of physical closeness. If you find these explorations calming or interesting, consider liking the video or subscribing. It helps this small corner of thoughtful biology reach others who might enjoy taking a slower look at how the human body shapes human experience. Now let's ease into the science behind something that feels deeply personal. When people fall in love, they often describe it as sudden, overwhelming, almost irrational. Heart rate increases, palms sweat, attention narrows around one person, thoughts circle back again and again. But those early sparks of attraction are not powered by oxytocin alone. In fact, at the beginning, other chemicals tend to take center stage. Dopamine surges in response to novelty and reward. Norepinephrine sharpens focus and alertness. Cortisol can rise, adding a slight stress response that makes everything feel heightened. Oxytocin enters more quietly. It does not create the rush of infatuation. Instead, it helps transform intense attraction into something steadier. It supports bonding. It reinforces closeness. It encourages trust and attachment. If dopamine is associated with pursuit and excitement, oxytocin is associated with staying. Biologically speaking, oxytocin is a peptide hormone composed of 9 amino acids. It is produced primarily in a region of the brain called the hypothalamus. From there, it travels down nerve fibers to the posterior pituitary gland, where it can be released into the bloodstream. It is also released directly within the brain, acting as a neurotransmitter that influences neural circuits tied to emotion and social behavior. This dual role, hormone in the body, messenger in the brain, is part of what makes oxytocin so interesting. It bridges physical processes and psychological experiences. Historically, oxytocin was first identified not as a love chemical, but as a hormone involved in childbirth. In the early 20th century, researchers discovered that extracts from the posterior pituitary gland could stimulate uterine contractions. The name itself comes from Greek roots meaning swift birth. Later, scientists found that oxytocin also triggers milk ejection during breastfeeding. For decades, its identity remained primarily tied to reproduction. It wasn't until the latter half of the 20th century that researchers began noticing something broader. Oxytocin levels rose not only during labour and nursing, but also during close social contact. In animal studies, certain species that formed long-term pair bonds showed distinct oxytocin patterns compared to those that did not. This observation opened the door to a new line of inquiry. Perhaps oxytocin played a central role in social attachment. One of the most influential animal models in this research involved prairie voles. Unlike many rodents, prairie voles tend to form monogamous pair bonds. After mating, they often remain close to one partner, sharing nests and raising offspring together. When scientists examined their brains, they found high densities of oxytocin receptors in areas linked to reward and reinforcement. In contrast, closely related species that did not form pair bonds showed different receptor patterns. By manipulating oxytocin levels or blocking its receptors, researchers could alter bonding behavior in these animals, increase oxytocin signaling and bonding strengthened, block it and attachment weakened. These findings did not prove that oxytocin single-handedly creates love, but they strongly suggested that it plays a supporting role in forming lasting social connections. Translating animal research to humans is always complex, but similar themes began to emerge. In human studies, oxytocin levels often rise during affectionate touch, prolonged eye contact, sexual intimacy, and even synchronized activities like singing or group rituals. Couples in the early stages of romantic attachment sometimes show elevated oxytocin concentrations compared to individuals who are single. Parents interacting warmly with their infants also exhibit measurable increases. What's particularly notable is how oxytocin interacts with the brain's reward circuitry. The ventral tegmental area and the nucleus accumbens, regions heavily involved in motivation and pleasure, respond to social bonding in ways that overlap with other rewarding experiences. Oxytocin appears to enhance the salience of social stimuli, making interactions with certain people feel more meaningful and reinforcing. This does not mean that oxytocin forces you to fall in love with whoever happens to be nearby. Context matters, personal history matters, psychological traits matter. The brain is not a vending machine where one chemical produces one outcome. Instead, oxytocin modifies the sensitivity of neural systems that evaluate social safety and connection. One important function of oxytocin is its role in reducing fear responses in certain situations. The amygdala, a brain structure associated with threat detection, often shows decreased activity in response to social cues when oxytocin levels are elevated. This reduction can promote a sense of calm and openness in trusted settings. When you feel safe with someone, when conversation flows without tension, part of that ease may be supported by oxytocin's modulation of threat perception. Safety is central here. Attachment at its core is about reducing vulnerability. Human infants are born highly dependent. Without caregivers, survival is impossible. Evolution favoured mechanisms that encourage proximity and bonding. Oxytocin became part of that system, reinforcing closeness between parent and child. Over time, those same circuits were adapted for adult pair bonding and social cooperation. Consider how early physical contact shapes development. Skin-to-skin contact between a parent and newborn triggers oxytocin release in both. This mutual increase supports bonding, stabilizes heart rate, regulates stress responses, and promotes long-term attachment patterns. The experience is biological before it is conceptual. A newborn does not understand love in words, but its nervous system recognizes safety and connection. As humans grow, oxytocin continues to influence social learning. Trust-building interactions, shared laughter, collaborative problem-solving, supportive conversation can reinforce oxytocin pathways. Repeated positive interactions strengthen associations between a particular person and feelings of security. Over time, this reinforcement helps shift relationships from uncertainty to reliability. Romantic relationships often follow a similar trajectory. Early attraction may be fueled by novelty and excitement. As partners spend time together, engage in physical affection, and share vulnerable experiences, oxytocin release can support the transition from infatuation to attachment. The relationship begins to feel less like a thrilling gamble and more like a stable bond. That said, oxytocin is not inherently good. It does not create universal kindness. Research shows that while oxytocin can increase trust within familiar groups, it can also heighten in-group favoritism. In some contexts, it may even amplify defensive responses toward perceived outsiders. Bonding is selective by design. Evolution favored strong attachments to specific individuals or groups, rather than indiscriminate trust toward everyone. So when we call oxytocin the chemical that makes you fall in love, we are simplifying a network of interactions. It is more accurate to say that oxytocin helps transform social contact into meaningful attachment. It lowers certain barriers, enhances perceived reward, and strengthens memory around bonding experiences. Memory is another important piece. Oxytocin interacts with hippocampal systems involved in encoding emotional memories. Moments of closeness can become deeply imprinted, reinforcing the value of a relationship. When you think back on a significant shared experience, a late-night conversation, a supportive gesture during a difficult time, the emotional weight of that memory may be partly shaped by the neurochemical environment in which it was formed. At the same time, individuals vary in their oxytocin systems. Genetics influence receptor density and sensitivity. Early life experiences shape attachment styles and stress responses. Some people may find it easier to trust and bond quickly. Others may approach closeness cautiously, with neural circuits shaped by different histories. Oxytocin operates within those personal frameworks. It does not erase them. In modern culture, it's tempting to reduce love to chemistry and chemistry to a single molecule. But love, as lived experience, involves cognition, memory, expectation, cultural norms, personal values, and shared narrative. Oxytocin supports the biological infrastructure that makes attachment possible. It does not replace the complexity of human choice and behavior. As we move deeper into this topic, it becomes helpful to separate three related but distinct processes: attraction, attachment, and long-term bonding. Each involves overlapping but distinct neural pathways. Oxytocin plays its most consistent role in attachment and bonding, rather than in the initial spark of desire. Understanding this distinction allows us to examine what actually happens in the brain during those early moments when someone begins to matter more than others. What shifts inside the neural circuitry? How do dopamine-driven reward loops transition into oxytocin-supported attachment networks? And why does one person, out of many, become uniquely significant? To answer that, we'll need to look more closely at the interplay between brain regions involved in motivation, emotion, and social evaluation. Falling in love feels sudden, but beneath that feeling lies a series of coordinated biological events, each influencing the next, gradually reshaping how one individual is represented in your internal world. When people describe falling in love, they usually talk about emotion first. They mention excitement, nervousness, distraction, anticipation. They talk about thinking constantly about one person. They describe physical sensations that feel almost involuntary. What's less obvious in the moment is that these experiences reflect measurable shifts inside the brain. Romantic attraction is not a vague cloud of feeling drifting through the mind. It is a coordinated biological state involving specific neural circuits, chemical messengers, and changes in attention, motivation, and behavior. The early stage of romantic love begins in systems that evolved long before modern relationships, long before dating culture, long before language itself. These systems are rooted in survival. They are designed to help organisms identify rewarding stimuli, pursue them, and remember them. When you meet someone who becomes romantically significant, those circuits become highly active. One of the first major players is dopamine. Dopamine is often simplified as the pleasure chemical, but that description is incomplete. Dopamine is more accurately associated with motivation, reward prediction, and goal-directed behavior. It does not simply make you feel good. It pushes you to seek, to pursue, to anticipate. When someone captures your romantic interest, dopamine activity increases in areas such as the ventral tegmental area, VTA, and the nucleus accumbens. These regions are central to the brain's reward pathway. Functional MR studies of individuals who report being intensely in love show strong activation in these dopamine-rich regions when they view a photograph of their romantic partner. The pattern resembles the neural response seen in other highly motivating experiences. The brain is not passively enjoying the presence of this person, it is actively orienting toward them as a goal. This is why early romantic attraction often feels urgent. The brain is tagging one individual as unusually significant. That tagging alters attention. You may find yourself scanning rooms for them, replaying conversations, checking messages more frequently than usual. Dopamine reinforces the expectation of reward, and that expectation sharpens focus. Alongside dopamine, norepinephrine also increases. Norepinephrine is linked to alertness, energy, and heightened memory encoding. Elevated norepinephrine contributes to the physical sensations often described during early attraction. Increased heart rate, sweaty palms, a sense of restlessness. It also strengthens memory for emotionally relevant details. That's why small moments, what someone was wearing, the exact phrasing of a sentence, the sound of their laugh, can feel unusually vivid in retrospect. Cortisol, a hormone associated with stress, may also rise in the early phase of romantic love. This might seem counterintuitive. If love is positive, why would stress hormones increase? The answer lies in uncertainty. Early attraction involves unpredictability. Will the other person reciprocate? How will this unfold? The brain responds to uncertainty with mild stress activation. That stress is not necessarily negative. In moderate amounts, it enhances attention and memory. It makes the experience feel important. Together, dopamine, norepinephrine, and cortisol create a state of focused motivation and heightened emotional salience. This is the phase people often describe as infatuation. Sleep may be disrupted, appetite may change. Thoughts circle repeatedly around the same person. The brain is prioritizing this potential bond. At this stage, serotonin levels can shift as well. Some research suggests that early romantic love is associated with reduced serotonin transporter availability, a pattern that resembles what is seen in obsessive-compulsive tendencies. This does not mean falling in love is a disorder. It does suggest that the brain temporarily enters a state of intensified preoccupation. Repetitive thinking about a romantic partner is not purely psychological. It reflects measurable neurochemical changes. While these chemical shifts are happening, specific brain regions involved in evaluation and social judgment begin to adjust. The prefrontal cortex, particularly areas involved in critical assessment, may show reduced activity when individuals view images of their romantic partner. In simpler terms, the brain can temporarily downregulate skepticism. Flaws become less salient. Positive attributes become amplified. This does not mean people become irrational. It means that the reward system can bias perception. If dopamine circuits are strongly activated, the brain interprets the stimulus, in this case a person, as valuable. Once something is coded as valuable, cognitive systems often align with that valuation. Meanwhile, the amygdala, which plays a role in processing fear and threat, may show altered responses in the presence of a trusted romantic partner. Early in attraction, anxiety can be elevated due to uncertainty. But as positive interactions accumulate, oxytocin begins to enter the picture more prominently. Oxytocin does not dominate the earlier sparks of attraction. Instead, it gradually increases during repeated positive social contact. Physical touch, prolonged eye contact, sexual intimacy, and emotional vulnerability can trigger oxytocin release from the hypothalamus. From there, it influences both the body and the brain. In the brain, oxytocin modulates activity in regions related to fear, reward, and social recognition. It can reduce amygdala reactivity in safe contexts, promoting a sense of calm around a specific person. It also enhances the perceived reward value of social interaction, reinforcing the bond. The more positive contact occurs, the stronger the association becomes. This shift marks a transition from pursuit to attachment. Dopamine may have initiated the chase, but oxytocin helps stabilize the connection. Over time, the neural representation of the partner becomes integrated into broader networks, involving memory, future planning, and identity. Another important structure involved in romantic love is the hippocampus, which supports memory consolidation. Shared experiences with a romantic partner are encoded with emotional significance. When oxytocin interacts with hippocampal circuits, it can strengthen the emotional tone of memories associated with closeness. This makes bonding moments more durable in recall. As attachment deepens, the brain's representation of the self can subtly expand. Studies have shown that individuals in close relationships may incorporate aspects of their partner into their self-concept. Neural responses to partner-related stimuli can overlap with regions activated during self-referential thinking. In practical terms, the boundary between me and us becomes less rigid. This integration is not purely symbolic. It reflects neural plasticity. Repeated shared experiences alter synaptic connections. The brain learns patterns of interaction, predicts responses, and builds expectations. Love, in this sense, is not just a feeling, but a form of learning. It's also important to note that not everyone experiences these processes in identical ways. Individual differences in attachment style, shaped partly by early caregiving experiences, influence how the brain responds to romantic closeness. Someone with a secure attachment pattern may show smoother transitions from attraction to bonding. Someone with an anxious attachment style may exhibit heightened amygdala reactivity and stronger stress responses during uncertainty. Someone with an avoidant pattern may show dampened reward activation in response to intimacy cues. Genetics also play a role. Variations in genes related to dopamine receptors, oxytocin receptors, and serotonin transporters can influence sensitivity to romantic cues. This does not determine destiny, but it can shape tendencies. Another factor is context. Cultural norms influence how relationships develop. Social expectations can alter how openly emotions are expressed, how quickly physical intimacy occurs, and how commitment is defined. The brain responds to lived experience within these frameworks. Biology provides mechanisms. Culture shapes how they are engaged. As romantic love matures, the neural profile shifts again. The intense dopaminergic spikes of early infatuation often stabilize. Brain imaging of long-term couples who report deep attachment still shows activation in reward pathways, but the pattern is less chaotic and more regulated. Oxytocin and vasopressin, another peptide involved in bonding, play stronger roles in maintaining long-term attachment. Vasopressin is particularly associated with pair bonding in some animal species, and appears to contribute to protective and affiliative behaviors in humans as well. While oxytocin is often emphasized, it operates within a broader network of bonding-related chemicals. At the same time, stress systems can become more regulated in stable relationships. When individuals perceive their partner as a reliable source of support, the hypothalamic pituitary adrenal (HPA) axis may show reduced reactivity during external stressors. In other words, having a trusted partner can buffer stress responses at a physiological level. This buffering effect illustrates how romantic love extends beyond emotion into health. Researchers linked supportive relationships with lower blood pressure, improved immune function, and longer lifespan. These associations are complex and influenced by many variables, but the biological interplay between bonding hormones and stress regulation likely contributes. It's also worth acknowledging that romantic love can activate pain circuits when threatened. Social rejection or breakup engages brain regions overlapping with those activated during physical pain. The anterior cingulate cortex and insula, involved in processing distress, show increased activity in response to romantic loss. The same systems that made the bond feel rewarding can make its disruption feel deeply uncomfortable. From an evolutionary perspective, this makes sense. If bonding increases survival probability through shared resources, cooperative child rearing, and mutual protection, then losing that bond represents a significant threat. The brain responds accordingly. Throughout all these stages, the brain remains plastic. Experiences reshape connections. Repeated positive interactions strengthen attachment pathways. Repeated conflict or betrayal can weaken them. Love is dynamic at the neural level. When people say they fell in love, the metaphor suggests a sudden drop. Neuroscience suggests something more layered. There is an initial spike in reward signaling. There is heightened attention and arousal. There is gradual reinforcement through repeated contact. There is memory consolidation, identity integration, stress modulation. None of these processes happen in isolation. They overlap and influence one another. Importantly, the brain does not experience romantic love in a vacuum. Other life stresses, mental health conditions, past traumas, and environmental pressures all interact with bonding circuits. A person under chronic stress may show different neurochemical patterns than someone in a stable environment. Sleep deprivation can alter dopamine sensitivity. Depression can dampen reward responses. The same neural systems involved in love are also involved in broader emotional regulation. Despite the complexity, certain patterns remain consistent. Early attraction activates reward pathways strongly. Attachment engages oxytocin-mediated bonding circuits. Long-term partnership integrates reward, memory, and stress regulation systems into a more stable configuration. Understanding these mechanisms does not reduce love to chemistry alone. It clarifies how biology supports emotional experience. The subjective richness of falling in love, anticipation, vulnerability, comfort, longing, emerges from coordinated neural activity. The brain does not manufacture meaning artificially. It encodes value through chemical signaling and circuit activation. As we continue examining oxytocin more closely, it becomes clear that its role is less about creating the initial spark and more about shaping what happens next. Attraction might draw two people together, but attachment determines whether they remain connected. To understand how that attachment solidifies, we need to look more carefully at how oxytocin is produced, how it travels through the brain, and how it alters the balance between fear, trust, and reward in ways that make closeness feel sustainable rather than overwhelming. When people first hear the term love hormone, it tends to create a clear mental image. A single chemical circulating through the body, responsible for affection, loyalty, bonding, maybe even romance itself. The nickname is simple and memorable. It suggests that love can be traced back to one measurable substance. But biology rarely works in single-cause explanations. Oxytocin is influential, yes. It is powerful in specific contexts, but it is not a switch labelled love that flips on when you meet the right person. Oxytocin is a peptide hormone made up of 9 amino acids. Chemically, that makes it relatively small compared to many proteins in the body. It belongs to a class of molecules that act as both hormones and neurotransmitters. That dual identity is important. As a hormone, oxytocin travels through the bloodstream to affect tissues in the body. As a neurotransmitter, it is released within the brain, where it influences neural communication directly. The production of oxytocin begins in the hypothalamus, a small but critical region located near the base of the brain. The hypothalamus regulates many essential functions: hunger, temperature, circadian rhythms, and hormone control. Within the hypothalamus, specific groups of neurons, primarily in the paraventricular nucleus and the supraoptic nucleus, synthesize oxytocin. From there, oxytocin molecules travel down long axons to the posterior pituitary gland. When triggered, they are released into the bloodstream. This pathway was identified long before oxytocin became associated with love or bonding. In fact, oxytocin was first studied in the context of childbirth. Researchers in the early 1900s observed that extracts from the posterior pituitary could stimulate uterine contractions in animals. The name oxytocin comes from Greek roots meaning swift birth. For decades, its primary medical use was in obstetrics. Synthetic oxytocin is still administered to induce or strengthen labour contractions and to reduce bleeding after childbirth. Another well-established function of oxytocin involves lactation. During breastfeeding, oxytocin triggers the milk ejection reflex. When an infant suckles, sensory nerves send signals to the hypothalamus, prompting oxytocin release. That release causes smooth muscle cells around milk-producing glands to contract, allowing milk to flow. This reflex is automatic, regulated by a feedback loop between physical stimulation and hormone release. So long before researchers connected oxytocin to romantic bonding, it was known as a hormone essential for reproduction and early maternal care. The transition from birth hormone to love hormone happened gradually as scientists began noticing its broader effects on social behaviour. In the latter half of the 20th century, animal research started to reveal patterns that extended beyond childbirth. Studies of certain mammalian species showed that oxytocin influenced maternal behaviour, grooming, proximity seeking, and pair bonding. The prairie vole became one of the most cited examples. Unlike many rodents, prairie voles form relatively stable pair bonds. When researchers examined their brains, they found dense distributions of oxytocin receptors in areas tied to reward processing. Receptors matter as much as the hormone itself. A hormone can circulate widely, but its effect depends on where receptors are located and how sensitive they are. Prairie voles have high concentrations of oxytocin receptors in the nucleus accumbens and prefrontal cortex, areas involved in reinforcement and decision-making. Closely related vole species that do not form strong pair bonds show different receptor patterns. By altering oxytocin signaling in prairie voles, either enhancing it or blocking it, scientists could modify bonding behaviours. These findings suggested that oxytocin was not simply a reproductive hormone. It appeared to link social interaction with reward circuitry. In other words, it helped make closeness reinforcing. As research shifted toward humans, measuring oxytocin became more complicated. Unlike controlled animal studies, human relationships involve layers of culture, memory, language, and expectation. Still, patterns began to emerge. Blood plasma samples showed elevated oxytocin levels in new couples reporting strong romantic attachment. Mothers interacting with their infants displayed increased oxytocin concentrations. Even activities such as hugging, holding hands, or maintaining prolonged eye contact could produce measurable increases. One of the reasons oxytocin influences social bonding lies in its effect on the amygdala. The amygdala plays a central role in detecting threat and processing fear. Elevated oxytocin can dampen amygdala reactivity in safe social contexts. This reduction does not eliminate caution entirely, but it lowers defensive barriers enough to make closeness feel manageable. When two people begin to trust each other, oxytocin helps shift the nervous system from guarded to receptive. At the same time, oxytocin interacts with dopamine systems. While dopamine drives pursuit and anticipation, oxytocin helps stabilize the reward associated with repeated social contact. This interaction is crucial. Without dopamine, attraction might not begin. Without oxytocin, attachment might not consolidate. It is also important to clarify that oxytocin does not operate uniformly across all social situations. It is context dependent. For example, experiments using intranasal oxytocin, where participants inhale a synthetic form through a nasal spray, have shown increased trust in certain structured tasks. In one well-known study, participants given oxytocin were more likely to invest money in a partner during a trust game. However, follow-up research revealed that this effect was not universal. Oxytocin increased trust primarily toward individuals perceived as belonging to the same social group. This finding complicates the idea of oxytocin as a universal empathy enhancer. Instead, it appears to strengthen social bonds selectively. Evolutionarily, this makes sense. Bonding tightly with trusted individuals improves cooperation and survival. Extending equal trust to strangers in all contexts could be risky. Beyond romantic and parental bonding, oxytocin influences a range of social behaviors. It plays a role in recognizing familiar faces. It affects vocal tone perception and emotional interpretation. It may contribute to synchrony during group activities such as singing or coordinated movement. In some studies, synchronized behavior among individuals, like rowing in unison or chanting, has been associated with increased oxytocin levels. The hormone also interacts with the stress response system. The hypothalamic pituitary adrenal axis regulates cortisol release during stress. Oxytocin can attenuate this response under supportive conditions. For example, when individuals experience a stressful task in the presence of a trusted partner, cortisol increases may be smaller compared to facing the same task alone. This buffering effect illustrates how social support translates into measurable physiological changes. However, oxytocin's effects are not always calming. In certain contexts, it may amplify emotional intensity rather than reduce it. For individuals with social anxiety or trauma histories, oxytocin can increase attention to social cues, which may heighten discomfort instead of easing it. This variability underscores that oxytocin does not create emotion from scratch. It modulates existing neural patterns. The distribution of oxytocin receptors in the brain further explains its wide influence. Receptors are found in the amygdala, hippocampus, nucleus accumbens, prefrontal cortex, and brainstem regions. This distribution allows oxytocin to influence fear processing, memory formation, reward evaluation, decision-making, and autonomic regulation. Genetics contribute to differences in receptor expression. Variations in the OXTR gene, which codes for the oxytocin receptor, have been associated with differences in social sensitivity, empathy, and attachment style. These associations are probabilistic rather than deterministic. They suggest that individuals vary in how responsive their oxytocin systems may be. Early life experiences shape these systems as well. Consistent caregiving in infancy can support healthy attachment patterns and balanced stress responses. Chronic neglect or unpredictable caregiving can alter stress regulation circuits and potentially influence how oxytocin interacts with those systems later in life. The hormone itself may be the same molecule, but its impact depends on the neural environment into which it is released. The popular media portrayal of oxytocin sometimes suggests that boosting it could solve social disconnection. Commercial products have even marketed oxytocin sprays as tools for enhancing relationships. Scientific evidence does not support such simple solutions. Intranasal oxytocin may temporarily influence certain behaviors under controlled conditions, but long-term relational health depends on patterns of interaction, communication, and mutual responsiveness. It is also worth distinguishing oxytocin from vasopressin, another peptide hormone produced in the hypothalamus. Vasopressin is structurally similar to oxytocin and plays roles in water balance and blood pressure regulation. In some animal studies, vasopressin has been linked to pair bonding and territorial behaviors, particularly in males. In humans, the interplay between oxytocin and vasopressin contributes to complex social behaviors, including attachment and protective instincts. Another common misunderstanding is that higher oxytocin always means stronger love. Hormone levels fluctuate naturally throughout the day. They respond to stimuli and then return toward baseline. Bonding is maintained not by a constant flood of oxytocin, but by repeated experiences that reinforce neural pathways. The hormone acts as a facilitator during key moments of contact. The timing of release also matters. Oxytocin surges during childbirth and breastfeeding are dramatic because they coordinate physiological processes critical for survival. In romantic or friendship contexts, the increases are generally smaller, but still meaningful within the brain signaling system. Neuroscientists sometimes describe oxytocin as enhancing the salience of social cues. Salience refers to how much attention the brain assigns to a stimulus. When oxytocin levels rise in a positive social setting, the brain may interpret that setting as especially relevant. Eye contact feels more significant. Touch feels more reassuring. Shared laughter feels more connecting. This heightened relevance strengthens learning about that person. Memory consolidation is part of this process. The hippocampus encodes emotionally significant events more robustly. When oxytocin interacts with memory circuits during bonding experiences, it helps embed those moments in long-term storage. Over time, a series of emotionally reinforced memories creates a narrative of connection. Importantly, oxytocin is not limited to romantic love. It plays roles in friendship, family bonds, group cohesion, and even pet ownership. Interactions between humans and companion animals have been shown to increase oxytocin levels in both. Mutual gaze between a dog and its owner, for example, can elevate oxytocin concentrations, reinforcing attachment across species. This broad involvement reflects oxytocin's evolutionary roots. Mammalian survival depends heavily on social structures. Cooperation in hunting, protection from predators, and shared caregiving required neural systems that rewarded proximity and trust. Oxytocin became part of that architecture. When describing oxytocin as the love hormone, the phrase captures one aspect of its function but obscures its range. It is more accurate to think of oxytocin as a bonding modulator. It influences how safe social closeness feels. It adjusts the balance between vigilance and openness. It strengthens the link between contact and reward. Understanding this complexity sets the stage for exploring how oxytocin is synthesized and released with such precision. Its journey from a cluster of neurons in the hypothalamus to widespread influence across the brain and body involves tightly regulated processes. To appreciate how a molecule composed of 9 amino acids can shape attachment patterns, we need to look more closely at the neural pathways that produce it and the triggers that prompt its release. Deep inside the brain, beneath the folds of the cortex and just above the brainstem, sits a structure no larger than an almond. It does not look dramatic in medical illustrations. It does not dominate the brain's surface the way the cortex does. Yet this small region regulates hunger, thirst, body temperature, sexual behavior, circadian rhythms, and much of the endocrine system. It is called the hypothalamus, and without it, attachment as we experience it would not unfold the way it does. Oxytocin begins here. The hypothalamus acts as a bridge between the nervous system and the endocrine system. It receives signals from across the brain, emotional cues, sensory input, internal physiological states, and translates them into hormonal instructions. Within the hypothalamus are clusters of specialized neurons that synthesize oxytocin. The two most important nuclei involved are the paraventricular nucleus, PVN, and the supraoptic nucleus, SON. These neurons are unique. Unlike many neurons that communicate only through synapses within the brain, oxytocin-producing neurons extend long axons downward into the posterior pituitary gland. This structure sits just below the hypothalamus and functions as a release site into the bloodstream. When these neurons fire, oxytocin travels along their axons and is secreted directly into circulation. At the same time, oxytocin is also released within the brain itself. Some of the same hypothalamic neurons send projections to other brain regions, including the amygdala, hippocampus, nucleus accumbens, and brainstem. This means oxytocin can influence peripheral organs and central neural circuits simultaneously. The synthesis of oxytocin begins at the genetic level. Within oxytocin neurons, the OXT gene is transcribed into messenger RNA. That RNA is translated into a precursor protein called preprooxytocin. This precursor includes oxytocin itself and a carrier protein known as neurophysin-1. As the molecule is processed within the cell, it is cleaved into active oxytocin and packaged into vesicles for transport. These vesicles travel down the axon toward release sites. The process is continuous but tightly regulated. Oxytocin is not constantly flooding the system. Its release depends on electrical activity in the producing neurons, which in turn depends on incoming signals from other parts of the brain. This is where attachment begins to connect to circuitry. The hypothalamus receives input from areas involved in emotion and social evaluation. The amygdala signals potential threat or safety. The prefrontal cortex contributes contextual interpretation and decision-making. Sensory regions transmit information about touch, facial expression, voice tone, and proximity. When a social interaction is interpreted as positive and safe, these signals can stimulate oxytocin neurons. Touch is one of the most reliable triggers. Gentle physical contact activates mechanoreceptors in the skin. These receptors send signals through the spinal cord to the brainstem and onward to the hypothalamus. Under supportive conditions, this pathway contributes to oxytocin release. The same process occurs during sexual intimacy, childbirth, and breastfeeding, although in those contexts the stimulation is more intense. Eye contact can also play a role. Sustained mutual gaze activates social recognition circuits in the brain, including areas that project toward the hypothalamus. This is one reason why prolonged eye contact can feel intimate. It engages neural pathways that influence oxytocin release. The release mechanism itself involves electrical firing patterns. Oxytocin neurons can fire in bursts. During childbirth, for example, these bursts become synchronized across many neurons, producing strong pulses of hormone release. In social bonding contexts, the firing patterns are typically more moderate, but still coordinated enough to alter neural communication. Once oxytocin enters the bloodstream, it travels to peripheral targets. In reproductive contexts, it binds to receptors in the uterus or mammary glands. In non-reproductive contexts, circulating oxytocin may influence heart rate, gastrointestinal function, and aspects of autonomic regulation. However, much of its role in attachment depends on its action within the brain. Oxytocin receptors are G-protein-coupled receptors. When oxytocin binds to them, it initiates intracellular signaling cascades that alter neuronal excitability. In some regions, oxytocin can reduce inhibitory signaling, making certain circuits more active. In others, it dampens activity, particularly in fear-related pathways. The amygdala is a key target. Under threat, the amygdala heightens vigilance. Oxytocin can reduce amygdala activation in safe social environments. This reduction does not eliminate the ability to detect danger. Instead, it fine-tunes the threshold at which social cues are interpreted as threatening. Around a trusted partner, this modulation makes openness possible. The nucleus accumbens is another important site. As part of the brain's reward system, it integrates dopamine signals related to motivation. Oxytocin enhances the salience of social stimuli in this region. When social interaction becomes rewarding, oxytocin helps reinforce that association. Repeated positive experiences strengthen synaptic connections, gradually embedding attachment into neural architecture. The hippocampus, responsible for memory consolidation, also contains oxytocin receptors. Emotional experiences encoded in the presence of elevated oxytocin may carry stronger affiliative tone. This contributes to the formation of meaningful relational memories. The hypothalamus does not operate independently. It is embedded within feedback loops. When oxytocin is released and social interaction proceeds positively, reduced stress responses can feed back to the hypothalamus, stabilizing its activity. Conversely, if social interaction is stressful or unpredictable, cortisol levels rise, which can alter hypothalamic function. Cortisol and oxytocin interact in complex ways. Acute stress can suppress certain aspects of social bonding. However, in supportive contexts, oxytocin can buffer cortisol release. The hypothalamus plays a central role in coordinating both systems. It regulates the hypothalamic pituitary adrenal (HPA) axis, which governs cortisol secretion. This means the same structure involved in attachment chemistry also manages stress responses. This integration explains why attachment feels stabilizing. When you're physically close to someone you trust, your heart rate may slow, breathing may deepen, and muscle tension may decrease. These physiological shifts reflect coordinated signaling between oxytocin pathways and autonomic control centers. The quiet nature of this chemistry is important. Unlike adrenaline, which produces rapid and obvious changes, oxytocin often works subtly. Its effects accumulate through repetition. A single positive interaction may produce a transient increase. Repeated experiences create lasting neural adjustments. Developmental timing matters as well. During infancy, the hypothalamus is highly responsive to caregiving input. Skin-to-skin contact, consistent soothing, and predictable responses from caregivers strengthen oxytocin-related circuits. These early experiences help calibrate the balance between vigilance and trust. Inconsistent or neglectful caregiving can alter this calibration, shaping attachment styles later in life. The hypothalamus remains plastic across the lifespan, but early patterns influence later sensitivity. This does not mean attachment capacity is fixed in childhood. Adult experiences can modify neural circuits. Stable, supportive relationships can gradually reshape stress regulation and bonding responses. Sex differences in oxytocin signaling have also been observed, though they are nuanced. Estrogen can increase oxytocin receptor expression in certain brain regions, which may influence responsiveness. However, both men and women produce oxytocin and rely on it for attachment. Cultural narratives that frame oxytocin as exclusively maternal oversimplify its function. Another layer involves neural synchrony. During close interaction, brain activity between partners can become synchronized in measurable ways. This synchrony correlates with oxytocin levels in some studies. Coordinated neural patterns may reinforce mutual understanding and emotional alignment. The hypothalamus participates indirectly in these patterns by modulating the neurochemical environment in which they occur. It is also worth noting that oxytocin neurons can release the hormone not only from axon terminals, but also from dendrites within the hypothalamus itself. This local release allows oxytocin to influence neighboring neurons, amplifying or regulating firing patterns. The result is a tightly controlled, context-sensitive system. Laboratory research has attempted to isolate oxytocin's effects using intranasal administration. While this method can influence behavior under certain conditions, it does not replicate the complexity of natural release patterns. Endogenous oxytocin release is tied to sensory input, emotional interpretation, and relational context. The hypothalamus integrates these signals in ways that cannot be fully reproduced by external dosing. The quiet chemistry of attachment unfolds over time. Each interaction feeds back into the system. Positive contact lowers threat perception, enhances reward signaling, and strengthens memory associations. The hypothalamus coordinates these changes without conscious awareness. From a subjective standpoint, attachment feels like comfort, safety, familiarity. From a physiological standpoint, it reflects repeated cycles of neural firing, receptor binding, synaptic plasticity, and hormonal modulation. The hypothalamus stands at the center of this coordination. As relationships deepen, oxytocin release patterns may become more predictable in response to specific cues. The voice of a partner, a familiar scent, a habitual gesture. These stimuli can activate hypothalamic neurons through learned associations. The body prepares for closeness even before conscious thought catches up. This predictive capacity is part of what makes long-term bonding feel stable. The nervous system anticipates safety in the presence of a trusted individual. That anticipation reduces baseline stress and supports cooperative behavior. Understanding the hypothalamus in this context shifts the image of oxytocin from a romantic abstraction to a regulated biological process. Attachment is not a sudden chemical flood. It is a coordinated interaction between sensory input, emotional evaluation, and hormonal response. The hypothalamus interprets signals and adjusts internal chemistry accordingly. From here, the next logical step is examining how physical contact, particularly touch, serves as one of the most consistent triggers for this system. The pathway from skin receptors to hypothalamic neurons illustrates how something as simple as a hug can influence neural circuits that shape trust and connection over time. Human skin is not just a protective covering. It is a sensory organ densely packed with receptors that translate physical contact into electrical signals. Every time someone brushes your arm, holds your hand, or places a reassuring hand on your shoulder, specialized cells embedded in your skin detect pressure, temperature, vibration, and stretch. Those signals travel through peripheral nerves into the spinal cord and upward toward the brain. What feels like a simple gesture at the surface is, underneath, a rapid exchange of electrical activity moving through highly organized pathways. Touch is one of the most direct ways to activate oxytocin-related systems. It does not require language. It does not require complex interpretation. It engages neural circuits that evolved long before verbal communication became central to human bonding. To understand why touch is so powerful in shaping trust, it helps to look closely at how these signals move from the skin to the hypothalamus and beyond. Within the skin, there are several types of mechanoreceptors. Merkel cells respond to sustained pressure. Meissner corpuscles detect light touch and texture. Pacinian corpuscles are sensitive to vibration. Ruffini endings respond to stretch. In addition to these, there is a class of unmyelinated nerve fibers known as C-tactile afferents. These fibers are particularly interesting in the context of social touch. They respond optimally to slow, gentle stroking at skin temperature. The kind of touch often associated with affection activates these fibers strongly. When C-tactile afferents are stimulated, they send signals along small diameter nerve fibers that conduct impulses more slowly than the fast, myelinated fibers responsible for sharp tactile discrimination. These signals travel to the spinal cord and then ascend to the brainstem. From there, they project to regions such as the posterior insular cortex, an area involved in representing the emotional quality of bodily sensations. This pathway is distinct from the one used for identifying object texture or precise location. It is specialized for the affective dimension of touch. In other words, it helps encode whether touch feels pleasant, soothing, or socially meaningful rather than simply where it occurs. Once the brain interprets touch as positive and safe, signals can influence the hypothalamus. This influence may occur through direct projections from limbic structures such as the amygdala and insula, which process emotional valence. If the context is supportive, hypothalamic oxytocin neurons become more likely to fire. The release of oxytocin that follows is not an automatic consequence of any touch. Context matters. A handshake with a stranger may not produce the same response as a hug from a trusted partner. The brain continuously evaluates social cues. Is this interaction voluntary? Is it expected? Is it associated with prior positive experience? These interpretations shape the neurochemical response. When touch occurs within a trusted relationship, oxytocin release can alter neural dynamics quickly. It can reduce activity in the amygdala, lowering vigilance. It can enhance reward-related activity in the nucleus accumbens. It can promote parasympathetic nervous system activation, slowing heart rate and deepening breathing. These physiological changes create the subjective experience of calm closeness. The autonomic nervous system plays a central role here. It consists of two main branches, sympathetic and parasympathetic. The sympathetic branch mobilizes the body for action. The parasympathetic branch promotes rest and recovery. Positive social touch tends to enhance parasympathetic activity through pathways involving the vagus nerve. Increased vagal tone is associated with emotional regulation and social engagement. Oxytocin interacts with these systems. It can support parasympathetic dominance under safe conditions. This shift contributes to the sense of relaxation often felt during sustained affectionate contact. The body is not preparing for threat. It is calibrating toward connection. Trust emerges partly from this physiological pattern. When repeated interactions consistently lower stress responses rather than elevate them, the brain updates its internal model of the other person. Predictability reduces uncertainty. Reduced uncertainty lowers cortisol release. Lower cortisol allows oxytocin signaling to operate without interference from stress pathways. Laboratory studies have attempted to measure this interaction directly. In experiments where participants undergo mild stress tasks, such as public speaking, those who receive supportive touch from a partner beforehand often show lower cortisol responses compared to those who do not. Oxytocin levels measured in saliva or plasma sometimes rise during the supportive interaction. The pattern suggests that physical reassurance can prime the nervous system towards stability. Trust is not only emotional, it is computational. The brain constantly makes predictions about outcomes. If past contact has been associated with comfort, the prediction shifts towards safety. Each positive touch strengthens the probability estimate that future contact will also be safe. Oxytocin enhances the reinforcement of this learning process. At the synaptic level, repeated activation of oxytocin receptors can influence plasticity. Long-term potentiation, a mechanism by which synapses become more efficient after repeated stimulation, can be modulated by oxytocin in certain brain regions. This means the association between a particular person and a sense of calm can become neurologically embedded. Importantly, the absence of touch can have measurable effects as well. Prolonged social isolation has been linked to altered oxytocin signaling and heightened stress reactivity. In animal models, deprivation of maternal contact during early development can disrupt normal oxytocin receptor distribution. In humans, chronic loneliness correlates with changes in stress hormones and inflammatory markers. While many variables are involved, the lack of physical social contact appears to influence neurochemical balance. The importance of touch is visible early in life. Newborn mammals rely on contact for regulation. Skin-to-skin contact between parent and infant stabilizes body temperature, heart rate, and glucose levels. It also promotes oxytocin release in both. These early cycles of co-regulation teach the infant's nervous system how to transition from distress to calm. The hypothalamus becomes attuned to signals of safety. As individuals grow older, touch remains significant, though cultural norms may alter its expression. Some societies encourage frequent physical contact among friends and family, others restrict it. Despite these differences, the underlying neural machinery remains similar. When touch is interpreted as supportive, it activates pathways that reinforce bonding. There are also differences in how individuals respond to touch. Personal history shapes perception. Someone with a background of inconsistent caregiving or trauma may experience heightened vigilance in response to physical proximity. In such cases, touch may initially increase amygdala activity rather than decrease it. Over time, consistent positive experiences can recalibrate this response, but the process may require repetition and stability. The specificity of oxytocin's effect becomes clearer when considering in-group and out-group dynamics. Research indicates that oxytocin can increase trust toward familiar individuals, while not necessarily extending that trust universally. Touch within a defined relationship reinforces that specific bond. It does not automatically generalize to all social contexts. Neuroimaging studies provide further insight. When individuals hold hands with a romantic partner during a stressful experience, activity in pain-related brain regions can decrease compared to holding the hand of a stranger or no one at all. The degree of reduction often correlates with reported relationship quality. This suggests that the meaning attached to touch influences neural processing. At a microscopic level, the electrical signals triggered by mechanoreceptors are converted into patterns of neurotransmitter release. Glutamate, GABA, dopamine, and oxytocin all interact within these networks. The balance among excitatory and inhibitory inputs determines whether the overall experience shifts toward calm engagement or defensive withdrawal. The speed of these processes can create the impression that trust develops instantly. In reality, each interaction modifies synaptic weights incrementally. Trust accumulates through repetition. The tiny electrical signals that begin in the skin contribute to larger-scale network changes over time. There is also evidence that synchronized touch can enhance social cohesion in groups. Activities such as dancing, team sports, or coordinated rituals often involve physical proximity and rhythmic movement. These shared experiences can elevate oxytocin and promote feelings of unity. The neural mechanisms overlap with those activated in one-to-one bonding, though the context differs. Touch is not the only pathway to oxytocin release, but it is one of the most reliable. Verbal affirmation, shared vulnerability, and mutual gaze also contribute. However, physical contact bypasses certain layers of cognitive interpretation. It engages sensory systems directly. In modern environments, physical contact can be limited by social structure, work patterns, and digital communication. While text messages and video calls can support emotional connection, they do not activate the same mechanoreceptor pathways. This difference does not invalidate digital relationships, but it highlights the unique biological role of physical presence. When people describe feeling reassured by a hug, they are reporting the subjective side of a measurable physiological shift. Heart rate variability increases, muscle tension decreases, oxytocin receptors bind circulating molecules, amygdala activity adjusts, the nervous system recalibrates. Trust emerges not only from what is said, but from how the body responds during interaction. If proximity consistently aligns with lowered stress and enhanced reward signaling, the brain updates its model of safety. Oxytocin supports this update by strengthening the neural pathways involved. Over time, repeated cycles of positive touch and reinforced trust can transition a relationship from tentative to stable. The electrical impulses that begin in the skin become part of a broader pattern of attachment chemistry. As these patterns solidify, bonding shifts from momentary sensation to enduring connection. This progression leads naturally into examining one of the most biologically intense contexts in which oxytocin and touch intersect: childbirth and early parent-infant bonding. In that setting, the coordination between physical contact, hormonal release, and attachment becomes even more pronounced, revealing how deeply embedded these systems are in human survival and development. Childbirth is one of the most physiologically intense events the human body undergoes. It involves coordinated muscle contractions, hormonal surges, shifts in cardiovascular function, and rapid changes in the newborn's environment. At the center of this orchestration is oxytocin. Long before it was associated with romance or trust experiments in laboratories, oxytocin was recognized as a hormone essential for labor. Its role in birth provides one of the clearest examples of how chemistry and attachment intersect from the very beginning of life. As labor approaches, oxytocin secretion increases in a pulsatile pattern. The hypothalamus sends signals to the posterior pituitary, releasing oxytocin into the bloodstream. This hormone binds to receptors in the uterine muscle, stimulating contractions. Those contractions push the fetus downward, stretching the cervix. Stretch receptors in the cervix send sensory information back to the brain, particularly to the hypothalamus. This feedback loop triggers additional oxytocin release. The cycle continues, intensifying contractions until delivery occurs. This process is known as a positive feedback loop. Unlike many hormonal systems that maintain balance through negative feedback, labor amplifies its own signals. Each contraction leads to more oxytocin, and more oxytocin strengthens contractions. The coordination must be precise. Too little oxytocin and labor stalls. Too much at the wrong time and the pattern becomes dysregulated. The body manages this timing through tightly controlled neural firing patterns in oxytocin-producing neurons. When the infant is delivered, the physiological demands shift quickly. The uterus continues contracting to prevent hemorrhage. At the same time, sensory stimulation from skin-to-skin contact and breastfeeding begins influencing oxytocin release in a different way. The context transitions from muscular effort to bonding. Immediately after birth, many hospitals encourage skin-to-skin contact between parent and infant. This practice is not only emotionally meaningful but biologically active. The newborn's skin temperature, heart rate, and breathing stabilize more effectively when placed against the parent's chest. Simultaneously, tactile stimulation activates mechanoreceptors that send signals to the parent's brain. Oxytocin levels rise again, but now in a bonding context rather than a contractile one. For the infant, the transition from the womb to the external environment is abrupt. Temperature changes, light exposure, gravity, and independent breathing all begin at once. Close physical contact provides regulation. Research has shown that infants who experience early skin-to-skin care often display more stable blood glucose levels and reduce stress markers compared to those separated for longer periods. Though the hormonal environment supports this adjustment, oxytocin released in the parent's brain can reduce amygdala activity and enhance reward circuitry, making attention toward the infant more focused and reinforcing. In functional imaging studies, parents viewing images of their own newborns show activation in dopamine-rich reward regions. Oxytocin likely contributes to strengthening these responses. Breastfeeding further amplifies the cycle. When the infant suckles, sensory nerves in the nipple transmit signals to the hypothalamus. Oxytocin neurons fire, releasing hormone into the bloodstream. This triggers the milk ejection reflex. Simultaneously, oxytocin released within the brain supports feelings of calm and bonding. Many parents report a sense of relaxation during nursing. That sensation reflects measurable shifts in autonomic activity and stress regulation. This interaction benefits the infant as well. Oxytocin is present in breast milk, though the extent to which it influences infant brain development directly is still being studied. What is clear is that the repeated cycle of feeding, contact, and soothing establishes predictable patterns. Predictability is central to secure attachment formation. Attachment theory, originally developed through psychological observation, aligns with these biological findings. Infants rely on caregivers not only for nutrition, but for regulation of arousal and emotion. When caregivers respond consistently to distress, through touch, vocal reassurance, feeding, the infant's nervous system learns to transition from activation to calm more efficiently. Oxytocin participates in this regulation on both sides of the interaction. The early postnatal period is often described as a sensitive window for bonding. This does not mean attachment cannot form later, or that brief separation prevents connection. It does suggest that hormonal and neural systems are particularly responsive during this time. The density of oxytocin receptors in certain brain regions may fluctuate across developmental stages, influencing sensitivity to social input. Research in animal models demonstrates that early tactile stimulation can shape long-term stress responses. For example, rodent pups that receive higher levels of maternal grooming show altered expression of stress-related genes and more regulated cortisol responses later in life. While direct parallels to humans must be drawn cautiously, similar principles appear to apply. Early caregiving experiences influence neurobiological development. In humans, parental mental health also affects this system. Postpartum depression has been associated with differences in oxytocin levels and altered neural responses to infant cues. Reduced oxytocin signaling may contribute to difficulties in bonding, though the relationship is complex and influenced by many factors, including prior mental health history, sleep deprivation, and social support. Importantly, bonding is not limited to biological mothers. Fathers, adoptive parents, and other caregivers also exhibit oxytocin increases during close interaction with infants. Studies measuring oxytocin in fathers during play and physical contact show similar patterns of elevation. The caregiving system is flexible. What matters most is consistent, responsive engagement. The first embrace after birth is often described emotionally, but it is also a measurable event. Heart rate variability shifts, cortisol levels adjust, oxytocin rises, neural circuits linking reward and social recognition activate. The newborn's cry, smell, and facial features all provide sensory input that feeds into these pathways. Olfaction plays a subtle role here. Humans are less reliant on smell than many mammals, but scent still contributes to recognition. Newborns can orient toward their caregiver's scent, and caregivers can often identify their infant by smell. This sensory channel interacts with limbic structures and may reinforce bonding through associative learning. Eye contact begins developing rapidly as well. Although newborn vision is limited at birth, the preference for faces emerges early. Mutual gaze strengthens over weeks and months. Each episode of eye contact triggers neural processing in social recognition circuits. Oxytocin may enhance the salience of these cues, reinforcing engagement. From an evolutionary perspective, the survival advantage of this system is clear. Human infants are highly dependent compared to many other mammals. They require prolonged caregiving. Mechanisms that reinforce caregiver motivation increase the probability of survival. Oxytocin helps ensure that caregiving behaviors are rewarding rather than purely effortful. This reinforcement extends beyond infancy. As children grow, physical affection, shared play, and emotional attunement continue to influence oxytocin release. Secure attachment patterns established early can support resilience in later relationships. Conversely, disrupted early bonding may influence vulnerability to anxiety or avoidance in adult intimacy. The biological processes involved in birth and bonding also highlight that oxytocin does not operate in isolation. Prolactin supports milk production. Endorphins modulate pain. Dopamine contributes to motivation and reward. Vasopressin influences protective behavior. The hormonal landscape is integrated, with oxytocin acting as one central component. Medical interventions can influence this landscape. Synthetic oxytocin is commonly administered during labor to induce or augment contractions. While it effectively stimulates uterine muscle, its central effects on bonding are less straightforward. Endogenous oxytocin released naturally in the brain may differ in pattern and timing from synthetic hormone delivered intravenously. Research continues to examine how these differences affect postpartum bonding, though findings are mixed and influenced by many variables. The broader point remains consistent. The same molecule that coordinates uterine contractions also shapes neural circuits underlying attachment. Its function evolves across contexts, from mechanical muscle stimulation to subtle modulation of trust and emotional closeness. In the hours and days following birth, repeated cycles of contact, feeding, soothing, and sleep reinforce attachment pathways. The infant learns that distress is followed by relief. The caregiver's brain learns to interpret subtle cues, changes in cry tone, facial expression, body movement. Oxytocin facilitates these learning processes by strengthening associations between contact and reward. Over time, the initial intensity of postpartum hormonal shifts stabilizes. Bonding transitions from acute hormonal surge to sustained relational pattern. The early chemistry sets the foundation, but ongoing interaction maintains the structure. Understanding birth and early bonding provides a reference point for later romantic attachment. The same neural circuits that once supported caregiver-infant closeness are recruited in adult pair bonding. The scale and context differ, but the underlying mechanisms overlap. Physical closeness, mutual regulation, shared safety. These elements echo the earliest attachment experiences. As bonding extends beyond infancy into broader social relationships, the patterns established through early contact influence how trust develops. To explore how these patterns translate into everyday adult interactions, it becomes useful to examine more subtle forms of touch and proximity that shape connection outside the intensity of childbirth. A handshake and a hug are both forms of physical contact. Both involve skin, pressure, proximity, and shared space. Yet most people instinctively recognize that they do not feel the same. A handshake is structured, brief, and socially formal. A hug is closer, longer, and typically reserved for people with whom we share some level of emotional familiarity. The difference in feeling is not simply cultural convention. It reflects differences in neural processing, hormonal response, sensory engagement, and context evaluation inside the brain. To understand why hugs feel different from handshakes, it helps to start with what happens at the level of the skin. The skin contains multiple classes of mechanoreceptors, each tuned to specific types of stimulation. When you shake someone's hand, the contact is usually firm and relatively short. Pressure-sensitive receptors such as Merkel cells and Meissner corpuscles activate quickly. These receptors provide detailed information about grip strength, movement, and texture. The signals travel rapidly along myelinated nerve fibers toward the somatosensory cortex, where the brain identifies the location and intensity of the contact. A handshake is processed largely as discriminative touch. It conveys information: how strong the grip is, how long it lasts, whether it is steady or hesitant. The primary neural pathway involved emphasizes precision and evaluation. Social interpretation follows quickly: confidence, professionalism, friendliness. But the tactile processing itself is focused on clarity and control. A hug, by contrast, engages a broader set of receptors and lasts longer. Slow, sustained pressure across the torso and shoulders activates not only the fast mechanoreceptors, but also C-tactile afferent fibers. These unmyelinated fibers respond best to gentle, prolonged stroking or pressure at skin temperature. Their signals travel more slowly and project to areas involved in emotional processing, particularly the insular cortex. The insula integrates bodily sensations with emotional awareness. When C-tactile fibers fire during a hug, the sensation is encoded not merely as pressure but as effective touch. The brain interprets the contact in terms of comfort, warmth, and safety, rather than simple physical information. This distinction contributes to the subjective difference between a brief greeting and a full embrace. Duration also matters. A typical handshake lasts only a few seconds. A hug often extends longer, sometimes beyond 20 seconds in close relationships. Studies have suggested that sustained physical contact increases the likelihood of measurable oxytocin release. The longer the contact remains safe and mutual, the more opportunity there is for hypothalamic oxytocin neurons to activate. Proximity amplifies this effect. During a handshake, individuals remain partially separated. Personal space is maintained. During a hug, bodies come into close alignment. Heart rates may synchronize slightly. Breathing patterns can influence one another. This physical alignment enhances autonomic regulation through the vagus nerve, supporting parasympathetic activation. The parasympathetic nervous system promotes relaxation and recovery. Increased vagal tone during supportive contact can lower heart rate and reduce cortisol levels. This shift is more likely during an embrace than during a brief formal gesture. The body interprets sustained closeness differently than a structured greeting. Context shapes the response further. A handshake is often used in professional settings, among acquaintances, or during introductions. It carries social meaning but limited emotional intimacy. The brain evaluates it within a framework of politeness and boundary maintenance. Amygdala activity related to vigilance may remain moderately engaged, particularly if the other person is unfamiliar. A hug, especially among trusted individuals, signals a reduction in social distance. It communicates willingness to share vulnerability. Because vulnerability carries risk, the brain must evaluate safety before fully relaxing. When the evaluation is positive, when the person is known and trusted, the amygdala reduces threat signaling. Oxytocin release may reinforce this safety assessment. The neurochemical environment during a hug differs from that of a handshake, partly because of expectation. If you anticipate warmth and comfort, the brain primes reward pathways. Dopamine and oxytocin interact in reinforcing the experience. A handshake rarely carries the same anticipatory emotional weight. Memory associations also play a role. Many people associate hugs with family, close friends, or significant others. These past experiences create neural links between hugging and positive emotional states. The hippocampus encodes these associations. When you enter a hug, the brain retrieves prior patterns shaping your current interpretation. Handshakes often carry associations of professionalism, negotiation, or introduction. These contexts activate different neural schemas. Even if the handshake is friendly, it rarely taps into early attachment circuits as strongly as a hug. Temperature exchange contributes subtly. During a hug, body heat is shared across a larger surface area. Thermoreceptors detect warmth, which can enhance feelings of comfort. Warmth has been shown in some psychological studies to influence perceptions of social closeness. The metaphor of warmth as kindness has physiological roots. Hormonal responses differ as well. While both forms of contact can produce minor shifts in stress hormones, sustained affectionate touch is more consistently linked to increases in oxytocin. Research measuring salivary oxytocin levels after prolonged hugs shows modest but detectable elevations, particularly among romantic partners or close family members. The predictability of touch also matters. A handshake follows a clear social script. Its boundaries are defined. A hug may vary in pressure, duration, and emotional tone. This variability can enhance its salience. The brain pays attention to socially meaningful variation, reinforcing learning. There are individual differences in how people respond. Attachment style influences perception. Individuals with secure attachment often experience hugs as calming and reassuring. Those with avoidant attachment may experience discomfort with prolonged closeness. In such cases, the same physical gesture can produce different amygdala activation patterns. Cultural background influences expectations as well. In some cultures, hugging is common among friends and acquaintances. In others, it is reserved for close family or romantic partners. The brain learns these norms over time. If hugging violates cultural expectations, it may activate vigilance rather than relaxation. Gender norms sometimes shape interpretation too, though biological responses themselves are not limited by gender. Social conditioning can influence who feels permitted to hug whom and in what settings. These expectations feed back into neural evaluation processes. It is also important to note that a hug is reciprocal. Both individuals must align their posture and pressure. This reciprocity fosters synchrony. Neural synchrony, patterns of coordinated brain activity between individuals, has been observed during close interaction. Although handshake interactions can involve synchrony, the sustained alignment of a hug may enhance this effect. From an evolutionary perspective, close physical contact played a role in survival. Early humans relied on group cohesion. Physical closeness reinforced alliance and caregiving. Hugs resemble the affiliative grooming behaviors observed in other primates. Grooming increases oxytocin levels in many species and strengthens social bonds. Handshakes do not have a clear analogue in early evolutionary history. They are relatively recent social conventions. Hugs and embraces likely predate formal greeting rituals by thousands of years. Pain perception also shifts differently between the two gestures. Studies have shown that holding a partner's hand during painful stimulation can reduce perceived pain intensity. A hug, involving more extensive contact, may amplify this buffering effect. The anterior cingulate cortex and insula, involved in processing pain and emotion, respond differently when supportive touch is present. These physiological changes reinforce why hugs feel subjectively deeper. They engage broader neural systems, somatosensory, emotional, reward, memory, autonomic regulation, and endocrine modulation. A handshake primarily engages evaluation and brief social acknowledgement circuits. This does not make one superior to the other. Each serves a social function. Handshakes facilitate boundary-respecting interaction. They allow individuals to signal goodwill without overstepping familiarity. Hugs communicate intimacy and reassurance when the relationship supports it. The distinction highlights how nuanced physical communication is. The body distinguishes not only what kind of touch occurs, but also who initiates it. how long it lasts, and what history exists between the individuals involved. Repeated hugs within a trusted relationship strengthen attachment pathways over time. Each instance reinforces safety learning. Handshakes rarely accumulate the same emotional weight unless they mark significant events. Understanding these differences clarifies how oxytocin operates selectively. It does not respond equally to all forms of contact. It is embedded in a system that weighs context, familiarity, duration, and reciprocity. The brain's interpretation determines whether attachment chemistry intensifies or remains neutral. This layered processing becomes even more visible when examining how attachment extends beyond individual touch to the hormonal balance between desire, bonding, and long-term commitment. Romantic love involves more than oxytocin alone. It includes interplay with other hormones that shape how attraction transforms into sustained partnership. Romantic attraction often feels unified. People describe being in love as if it is a single emotional state. Yet biologically, what we label as love includes at least two overlapping but distinct systems: lust and attachment. These systems share some neural pathways, but they are regulated by different hormonal patterns and serve different evolutionary purposes. Understanding the hormonal balance between lust and love helps clarify why desire can appear suddenly, why attachment develops more gradually, and why the two do not always move in perfect synchrony. Lust is primarily driven by sex hormones. In both men and women, testosterone plays a central role in sexual desire. Although commonly associated with male physiology, testosterone is present in all sexes and influences libido across the board. Estrogen also contributes to sexual motivation, particularly in women, where cyclical fluctuations can alter levels of interest and responsiveness. These hormones act on brain regions involved in sexual arousal, including the hypothalamus, amygdala, and parts of the limbic system. When testosterone levels rise, neurons in these areas become more responsive to sexual cues. Visual stimuli, scent, voice tone, and physical appearance can trigger activation in circuits associated with desire. The brain interprets these signals as opportunities for reproduction. Dopamine is heavily involved in this stage as well. Sexual attraction activates reward pathways in the ventral tegmental area and nucleus accumbens. This activation produces motivation and pursuit. The combination of dopamine and sex hormones creates a state characterized by heightened attention, anticipation, and physical arousal. Importantly, lust does not require deep familiarity. It can be triggered by novelty. The brain is wired to respond strongly to new potential mates, particularly when reproductive hormones are active. This novelty response explains why early attraction often feels intense and focused. Love, in contrast, relies more heavily on bonding hormones such as oxytocin and vasopressin. While lust prepares the body for sexual interaction, attachment prepares the brain for long-term affiliation. The neural emphasis shifts from pursuit to stability. After sexual intimacy, oxytocin levels often rise in both partners. In women, oxytocin release is associated with sexual climax and physical closeness. In men, vasopressin may also increase alongside oxytocin. These hormones contribute to post-intimacy bonding. The experience becomes linked not only to pleasure, but to the specific partner involved. This bonding process is reinforced by repetition. Each shared positive experience strengthens neural associations between that person and reward. Over time, dopamine-driven novelty gives way to oxytocin-supported familiarity. The reward shifts from excitement to reliability. The difference between lust and love becomes clearer when considering time course. Lust can ignite quickly, often within moments of visual or sensory stimulation. Attachment requires accumulation of interaction. It builds through shared experience, mutual vulnerability, and consistent regulation of stress. Neuroimaging studies illustrate this distinction. When individuals in early-stage passionate attraction view images of their partner, dopamine-rich areas show intense activation. As relationships mature, activation remains in reward circuits, but becomes less chaotic and more integrated with regions associated with long-term planning and emotional regulation. Oxytocin plays a role in reducing fear responses toward a specific partner. This reduction supports trust and emotional openness. Lust alone does not necessarily reduce threat perception. In fact, sexual attraction can coexist with anxiety or uncertainty. Attachment gradually stabilizes those fluctuations. Vasopressin adds another layer to this hormonal balance. Structurally similar to oxytocin, vasopressin has been linked to pair bonding in several animal species. In prairie voles, vasopressin receptor distribution influences male bonding behavior. In humans, vasopressin appears to contribute to protective and affiliative tendencies, particularly in long-term partnerships. The interplay between oxytocin and vasopressin may help explain why sexual intimacy can strengthen emotional bonds in established relationships. Repeated hormonal reinforcement supports exclusivity and loyalty in certain contexts. However, individual variation in receptor distribution and sensitivity means responses are not uniform. Sex hormones themselves can fluctuate within long-term relationships. Testosterone levels may decline slightly in men who enter committed partnerships and become fathers. Some studies suggest that reduced testosterone may correlate with increased nurturing behavior. In women, hormonal cycles continue influencing sexual interest even within stable attachments. This hormonal fluidity explains why lust and love can diverge. It is possible to experience strong sexual desire without deep attachment. It is also possible to maintain deep attachment even as sexual intensity changes over time. The systems are interconnected but not identical. From an evolutionary standpoint, separating lust from attachment had adaptive value. Lust promoted reproduction by motivating individuals to seek mates. Attachment promoted cooperative parenting and resource sharing, increasing offspring survival. The hormonal architecture reflects these dual pressures. Conflict between these systems can create emotional complexity. Novel attraction outside an established partnership may trigger dopamine and sex hormone responses without activating attachment circuits. Meanwhile, oxytocin-mediated bonding with a long-term partner continues operating. The brain can hold competing signals simultaneously. Long-term relationship satisfaction often depends on integrating these systems rather than relying exclusively on one. Sustained sexual interest benefits from novelty and variation, which can reactivate dopamine pathways. Emotional closeness benefits from consistent oxytocin-supported trust. When both systems receive stimulation, relationships tend to feel more balanced. Stress influences this balance as well. Chronic stress elevates cortisol, which can suppress sex hormone production and dampen libido. High stress can also interfere with oxytocin's calming effects. Under prolonged stress, both lust and attachment may weaken temporarily. This underscores the importance of environmental stability for relational health. Sleep and physical health also affect hormonal equilibrium. Sleep deprivation reduces testosterone levels and alters cortisol rhythms. Physical exercise can influence hormone production positively, supporting sexual health and mood regulation. The hormonal landscape is dynamic, shaped by lifestyle as well as relational factors. Age introduces additional changes. In early adulthood, sex hormone levels are typically higher, supporting strong libido. As individuals age, testosterone and estrogen levels may decline gradually. Attachment, however, often remains robust. In many long-term couples, emotional closeness persists even as sexual frequency changes. Oxytocin's role becomes particularly important during these later stages. Familiarity, shared history, and mutual regulation of stress sustain connection. The reward shifts from novelty-driven excitement to comfort-driven stability. The brain learns to value predictability. Cultural influences interact with biology here as well. Societal expectations about romance, fidelity, and gender roles shape how individuals interpret hormonal experiences. For example, a culture that idealizes constant passion may frame normal fluctuations in desire as failure. even though biologically they reflect typical transitions from lust to attachment. It is also worth recognizing that not all romantic relationships prioritize sexual intimacy equally. Some individuals experience low baseline libido but strong capacity for emotional attachment. Others may experience strong sexual drive with less inclination toward long-term bonding. Variations in hormone sensitivity and receptor expression contribute to these patterns. Neuroscience does not reduce love to chemicals, but it clarifies how different chemicals support different dimensions of connection. Testosterone and estrogen energize pursuit. Dopamine reinforces anticipation. Oxytocin and vasopressin stabilize bonds. Cortisol modulates stress within these systems. The transition from lust to love often involves recalibration of priorities within the brain. Early-stage attraction narrows focus intensely on one individual, driven by dopamine and novelty. As familiarity increases, the intensity may stabilize. Oxytocin supports this stabilization by reinforcing safety and trust. Importantly, attachment does not eliminate desire. In healthy partnerships, these systems can coexist. Sexual intimacy continues to trigger oxytocin release, which in turn reinforces bonding. The cycle becomes self-sustaining when positive experiences accumulate. Understanding this hormonal balance helps explain why relationships evolve over time. Initial passion may feel overwhelming. Long-term attachment may feel steady and grounding. Both states reflect coordinated neurochemical activity shaped by context, repetition, and individual differences. As we continue examining oxytocin's role in bonding, it becomes useful to look beyond human relationships and consider how similar hormonal systems operate in other species. Animal research, particularly in socially monogamous mammals, has provided some of the clearest insights into how attachment chemistry functions at a fundamental level. When scientists began trying to understand how bonding works in the brain, they did not start with humans. Human relationships are influenced by language, culture, memory, and personal history. That makes them difficult to isolate experimentally. Instead, researchers turned to animals whose social structures could be observed, measured, and compared under controlled conditions. One of the most important discoveries about attachment chemistry came not from studying people in love, but from studying small rodents living in grasslands. Not all animals form long-term pair bonds. In fact, across mammals, social monogamy is relatively rare. Many species mate with multiple partners across a breeding season, some form temporary bonds only during mating, others show no lasting association between partners once reproduction has occurred. Yet a few species display strong, selective, and enduring pair bonds. These species became central to understanding the biological foundations of attachment. The prairie vole is the most well-known example. Prairie voles are small rodents native to North America. Unlike many other vole species, prairie voles tend to form stable pair bonds after mating. Once bonded, they share nests, groom one another, and jointly care for offspring. They also show signs of partner preference, meaning they actively choose to spend time near their bonded mate rather than with unfamiliar individuals. In laboratory experiments, researchers observed that after mating, prairie voles displayed a marked shift in behavior. When given a choice between their partner and a stranger, they consistently preferred proximity to the partner. This preference could be measured objectively. Time spent in physical contact, frequency of grooming, and willingness to defend the partner from intruders all increased after bonding. When scientists examined the brains of prairie voles, they found unusually high densities of oxytocin receptors in regions associated with reward and reinforcement, particularly the nucleus accumbens and prefrontal cortex. They also found high densities of vasopressin receptors in areas related to social memory and territorial behavior. To test whether these receptors were merely correlated with bonding or actively driving it, researchers manipulated the system. When female prairie voles were given drugs that blocked oxytocin receptors, they failed to form partner preferences even after mating. When oxytocin was administered artificially in the absence of mating, partner preference could be induced. In males, blocking vasopressin receptors disrupted bonding behavior. These experiments demonstrated that oxytocin and vasopressin signaling were not incidental. They were functionally necessary for pair bond formation in this species. The story became even more compelling when researchers compared prairie voles with closely related species such as meadow voles and montane voles. These species do not form long-term pair bonds. They mate, but they do not show the same enduring partner preference. When their brains were examined, the overall levels of oxytocin were similar to prairie voles. The key difference lay in receptor distribution. In meadow voles, oxytocin receptors were less densely concentrated in reward-related brain regions. Without that receptor pattern, mating did not strongly link a specific partner to the reward system. The experience remained reproductive but did not transform into attachment. Further experiments showed that altering receptor distribution could shift behavior. When scientists genetically engineered voles to express more vasopressin receptors in bonding-related areas, they displayed increased affiliative behavior resembling monogamous patterns. This finding suggested that small changes in receptor placement could significantly influence social structure. These results reshaped how scientists thought about attachment. Bonding was not merely a learned social pattern. It was deeply tied to neurochemical architecture. The way oxytocin and vasopressin interacted with reward circuitry determined whether mating led to partnership. However, even in prairie voles, monogamy is not absolute. While they form strong pair bonds, occasional extra-pair mating does occur in natural environments. This highlights an important point. Biological predisposition does not guarantee rigid behavioral outcomes. Environmental pressures, population density, and resource availability all influence social patterns. Beyond voles, other species provide additional insight. Certain bird species, such as swans and albatrosses, form long-term pair bonds lasting multiple breeding seasons. In some cases, these bonds persist for life. While avian neurochemistry differs somewhat from mammals, analogous hormones and receptor systems support affiliative behavior. Among primates, gibbons are often cited as examples of socially monogamous mammals. Gibbon pairs maintain territories and coordinate care of offspring. While detailed neurochemical mapping in primates is more complex, oxytocin and vasopressin appear to play roles in maintaining these bonds. In contrast, species like chimpanzees exhibit more fluid mating systems. They form alliances and social hierarchies, but do not typically establish exclusive pair bonds. The distribution of bonding-related receptors in these species differs from those seen in more monogamous mammals. These cross-species comparisons reinforce that monogamy is not simply a moral or cultural construct. It is linked to measurable neurobiological differences. That does not mean humans are strictly monogamous or strictly non-monogamous by biology alone. It means that the capacity for pair bonding is supported by specific hormonal systems. In humans, oxytocin and vasopressin receptor distribution varies among individuals. Genetic variations in receptor genes have been associated with differences in relationship stability, empathy, and attachment style. Some studies have found correlations between certain vasopressin receptor gene variants and marital satisfaction in men. These associations are subtle and influenced by many other factors, but they suggest parallels with animal findings. It is important to distinguish between social monogamy and sexual monogamy. Social monogamy refers to forming a stable partnership for cooperative living and child-rearing. Sexual monogamy refers to exclusivity in mating. In many animal species, social monogamy does not guarantee sexual exclusivity. The neurochemical systems primarily support social bonding rather than strict reproductive exclusivity. From an evolutionary perspective, social monogamy can offer advantages in environments where offspring require extensive care. Human infants are highly dependent for many years. Cooperative parenting increases survival probability. Pair bonding, supported by oxytocin and vasopressin, may have provided evolutionary benefits by encouraging long-term parental investment. At the same time, humans possess flexible social strategies. Anthropological research shows a wide range of mating systems across cultures, from strict monogamy to polygyny and other arrangements. The presence of bonding hormones does not lock behavior into one rigid pattern. Instead, it provides a biological capacity for attachment that interacts with culture and environment. Animal research also reveals that bonding involves more than just hormones. Social memory is critical. In prairie voles, the ability to recognize and remember a partner supports selective attachment. Oxytocin enhances social recognition in several species, including humans. Without accurate social memory, pair bonding would be less stable. Another factor is stress buffering. In bonded animals, the presence of a partner reduces physiological stress responses. Cortisol levels may decline when a mate is nearby. This stress reduction reinforces the bond because proximity becomes associated with comfort and regulation. Territorial defense can also be influenced by bonding hormones. In some species, vasopressin is associated with increased protective behavior toward a mate or offspring. This does not necessarily translate to aggression universally, but it highlights how bonding chemistry can shape protective instincts. While prairie voles have become the most cited example in attachment research, scientists caution against oversimplifying. No single species perfectly models human relationships. Humans possess language, abstract thought, and cultural institutions that add layers of complexity. However, the fundamental observation remains. Specific neurochemical systems link social contact to reward and reinforcement. Understanding monogamy in animals clarifies that attachment is not accidental. It is supported by neural circuitry that can vary in structure and sensitivity. Small changes in receptor density can shift social behavior patterns dramatically in certain species. This insight returns us to the broader theme of oxytocin's role in human bonding. If receptor placement and hormonal interaction can influence whether an animal forms lasting partnerships, then variations within human neurobiology may contribute to differences in how individuals experience attachment, loyalty, and intimacy. At the same time, humans are not bound by receptor maps alone. Cognitive appraisal, personal values, and social context all influence relationship choices. The hormonal system provides capacity, not destiny. Animal studies have provided a controlled window into how bonding chemistry operates at a fundamental level. By observing how oxytocin and vasopressin shape partner preference in simpler systems, researchers gained tools for investigating similar processes in humans. The next step is examining whether it is possible to manipulate this chemistry directly, and what happens when scientists attempt to influence attachment artificially. Once scientists established that oxytocin plays a measurable role in bonding, a natural question followed. If this hormone helps reinforce attachment, can it be used to create it? If oxytocin strengthens trust and closeness, could administering it artificially enhance relationships, repair social difficulties, or even generate feelings of love where none existed before? The idea is appealing because it suggests a direct lever inside the brain. Increase oxytocin, increase connection. But biological systems rarely function in such linear ways. Manufacturing love through chemistry turns out to be far more complicated than increasing a hormone level. The most common method researchers have used to study oxytocin's effects in humans is intranasal administration. Participants inhale a mist containing synthetic oxytocin, which is believed to travel along olfactory and trigeminal pathways into the brain. From there, it may influence neural activity in regions rich in oxytocin receptors. This method does not flood the bloodstream in the same way intravenous administration would. Instead, it aims to bypass the blood-brain barrier partially. Even so, the exact amount that reaches central brain structures remains debated. Measuring oxytocin directly in the brain of living humans is not currently feasible, so much of the evidence relies on behavioral changes observed after administration. Early experiments produced intriguing results. In one widely cited study, participants who received intranasal oxytocin were more likely to invest money in a partner during a trust-based economic game. Compared to a placebo group, they displayed greater willingness to risk betrayal. This finding led to headlines suggesting that oxytocin could increase trust. Subsequent research showed that the effect was more nuanced. Oxytocin did not universally increase trust toward everyone. It appeared to enhance trust selectively, particularly toward individuals perceived as part of the same social group. In some cases, it even strengthened defensive bias against perceived outsiders. Rather than creating indiscriminate goodwill, oxytocin amplified existing social boundaries. Other studies explored oxytocin's influence on empathy and emotional recognition. Participants given oxytocin sometimes showed improved ability to interpret facial expressions or increased attention to eye regions during conversation. However, these effects were modest and often dependent on baseline personality traits. For individuals with certain social processing difficulties, such as those on the autism spectrum, researchers investigated whether oxytocin could enhance social responsiveness. Some short-term studies reported slight improvements in eye contact or emotional interpretation. Larger and longer-term trials, however, produced mixed results. The hormone alone did not fundamentally reshape social behavior. Romantic relationships became another area of interest. Could oxytocin increase feelings of closeness between partners? In controlled experiments, couples who received intranasal oxytocin before engaging in structured discussions sometimes displayed more positive communication patterns. They maintained eye contact longer or showed reduced physiological stress responses during conflict. Yet these effects were typically short-lived. Once the immediate pharmacological influence faded, relationship dynamics returned to baseline patterns. Oxytocin did not override underlying issues such as unresolved conflict, mistrust, or incompatibility. It could modulate momentary responses but not rewrite relational history. This limitation reflects how attachment works. Bonding depends on repeated interaction, memory consolidation, and reinforcement of shared experiences. A single chemical boost cannot replace accumulated trust. The brain's reward and memory systems require consistent input to alter long-term attachment pathways. There are also individual differences in response to oxytocin administration. Variations in the OXTR gene influence receptor sensitivity. People with certain receptor variants may respond more strongly to intranasal oxytocin, while others show minimal change. Psychological factors also matter. Individuals with secure attachment patterns may experience calming effects. Those with high social anxiety may become more vigilant to social cues instead. Context remains critical. In supportive environments, oxytocin can enhance feelings of safety. In competitive or ambiguous settings, it may heighten social awareness without necessarily increasing comfort. The hormone amplifies salience rather than dictating direction. Another misconception is that oxytocin produces pleasure directly. Unlike dopamine, which strongly activates reward circuits associated with euphoria and motivation, oxytocin's effects are subtler. It enhances the perceived significance of social stimuli, but does not generate intense excitement on its own. Ethical concerns arise when discussing chemical manipulation of attachment. If it were possible to artificially enhance bonding, questions about consent and autonomy would follow. Could such interventions be misused in coercive settings? At present, the limitations of oxytocin's effects make such scenarios unlikely, but the ethical discussion remains relevant. Commercial interest in oxytocin has led to the marketing of sprays claiming to enhance attraction or improve relationships. Scientific evidence does not support these claims as reliable or lasting. The social brain operates through complex networks shaped by history and experience. There is no shortcut to sustained attachment. Long-term changes in bonding likely require structural neural adaptation, synaptic plasticity, receptor regulation, and network connectivity shift gradually in response to repeated interaction. While oxytocin facilitates these processes during natural bonding, artificially introducing the hormone without corresponding experience does not create meaningful relational change. Stress modulation illustrates this principle. Oxytocin can buffer cortisol responses temporarily. However, chronic stress caused by ongoing relational conflict cannot be resolved by hormonal adjustment alone. Behavioral change, communication, and environmental stability are necessary to shift long-term stress patterns. Research has also examined whether oxytocin could reduce jealousy or increase fidelity. Findings remain inconclusive. Some studies suggest oxytocin may increase perceived attractiveness of a current partner while decreasing attention to alternative partners, particularly in men who are already committed. This effect appears to reinforce existing bonds rather than create new ones. It is important to distinguish between enhancing attachment within a relationship and manufacturing love where none exists. Oxytocin appears more capable of strengthening established bonds than initiating attachment without prior emotional investment. Neuroscience supports this distinction. During natural bonding, oxytocin release coincides with dopamine-driven reward learning. The partner becomes associated with positive emotional states. If oxytocin is administered without corresponding rewarding experience, the associative learning component is weak or absent. Animal studies reinforce this point. In prairie voles, oxytocin administration can induce partner preference, but typically in conjunction with mating or close contact. Without social interaction, the hormone alone does not generate stable attachment behavior. The idea of manufacturing love also overlooks the cognitive dimension of relationships. Humans evaluate compatibility, shared values, long-term goals, and personal boundaries. These evaluations involve prefrontal cortex activity and complex reasoning. Oxytocin does not override conscious judgment entirely. Moreover, attachment is bidirectional. Even if one individual experienced increased bonding chemistry, the relationship would not stabilize without mutual engagement. Love involves reciprocal reinforcement across two nervous systems. The broader lesson from research is that oxytocin acts as a modulator rather than a creator. It adjusts neural sensitivity to social cues. It enhances the weight assigned to particular interactions. It lowers barriers under safe conditions, but it operates within the context of lived experience. Attempts to pharmacologically engineer attachment reveal how deeply relational processes depend on repetition and meaning. The brain requires coherent narrative, consistent interaction, and shared history to build lasting bonds. This does not mean chemical influence is irrelevant. In clinical contexts, understanding oxytocin's role may inform treatments for social disorders or trauma-related attachment difficulties. Combined with therapy, environmental support, and behavioral practice, hormonal modulation could complement broader interventions. Alone, it is insufficient. Manufacturing love suggests a mechanical process. The reality is more distributed. Attachment emerges from coordinated activation of sensory systems, memory networks, reward circuits, and stress regulation pathways. Oxytocin is part of that coordination, but not its sole architect. Recognizing these limitations leads to a more grounded understanding of bonding chemistry. Rather than asking whether love can be manufactured, it becomes more useful to examine how trust and cooperation develop naturally in structured social interactions, and how oxytocin influences those processes in measurable ways. The idea of trust in a bottle is an intriguing concept. Imagine a substance that could take the uncertainty of human relationships and transform it into something more predictable, more reliable, more positive. Oxytocin, often called the trust hormone, has been at the center of this idea, thanks to its ability to influence social behavior and interactions. But while the notion of bottling trust is appealing, the reality is more complex. Trust is not merely a biological response to a single chemical. It is shaped by an intricate blend of neurochemistry, personal experience, social context, and cognitive processing. Oxytocin plays a key role, but it does not act alone. The psychological concept of trust involves the willingness to be vulnerable to others, to rely on them, and to expect that they will not cause harm. Trust is foundational in all relationships, romantic, familial, professional, and even in casual interactions. It is built through repeated positive experiences, shared vulnerability, and consistency over time. It is also influenced by individual and cultural differences. Understanding the role of oxytocin in trust, then, requires not just to look at the chemical processes involved, but also the cognitive and emotional factors that guide how trust develops. Oxytocin is released in response to social cues that signal safety and connection. One of its most well-known roles is in facilitating bonding between parents and their infants. When a mother gazes at her baby, holds it close, or breastfeeds, oxytocin is released in both the mother's and the baby's brain. This hormone enhances the feelings of attachment and trust that are vital for the infant's survival and for the continued care that the mother provides. However, the role of oxytocin extends beyond the parent-child bond. It is involved in the formation of trust in adult relationships as well. In laboratory settings, oxytocin has been shown to increase trust in economic games, where participants are asked to decide how much money to invest in a partner, with the understanding that the money may or may not be returned. When participants receive intranasal oxytocin, they are more likely to trust a partner, meaning they invest more money without knowing whether they will get it back. This has led to the popular idea that oxytocin makes people more trusting in general. While these findings are compelling, the relationship between oxytocin and trust is more nuanced. Research suggests that oxytocin does not make people universally trusting. Instead, it amplifies trust toward individuals who are already perceived as trustworthy or part of a familiar group. In other words, oxytocin may enhance the feelings of trust and connection we already have with certain people, but it does not automatically extend that trust to strangers or those who have demonstrated untrustworthy behavior. This selective effect of oxytocin on trust is key to understanding how it functions in social interactions. In an experiment where participants received oxytocin before playing a trust game, the increase in trust was not universal. Individuals were more trusting toward those they already knew or liked. Oxytocin did not erase skepticism or wariness of unfamiliar people. In fact, some studies have shown that oxytocin can increase in-group favoritism and out-group suspicion. For example, when oxytocin was administered to participants, they were more likely to show trust and generosity toward individuals who shared the same social identity, such as nationality or political affiliation, but less so toward those who were outside their group. This finding is significant because it helps explain why oxytocin is not a universal trust booster. It does not simply reduce fear of strangers or enemies. Instead, it fine-tunes social perception. Oxytocin helps individuals navigate their social world by increasing trust within familiar groups or in relationships where trust has already been established. In this way, oxytocin plays a role in the development of social bonds rather than the indiscriminate spreading of trust. The role of oxytocin in trust also highlights its connection to empathy. Empathy is the ability to understand and share the feelings of another person. Trust and empathy are closely linked, because in order to trust someone, you must believe that they understand and respect your feelings, and vice versa. Oxytocin has been shown to increase empathy in certain contexts. In one study, participants given oxytocin were better able to interpret emotional expressions, particularly in facial expressions and tone of voice. This enhanced ability to read social cues can make it easier to build trust, because it helps individuals respond to each other's emotions accurately. Oxytocin's influence on empathy also explains why it might strengthen relationships. When you feel that someone understands you emotionally and responds with care, trust can deepen. This emotional resonance helps create a stable foundation for both trust and attachment. Oxytocin, therefore, helps strengthen the emotional bonds that underlie long-term relationships, whether romantic, familial, or friendships. However, it is important to recognize that oxytocin's effects are not always purely positive. In some situations, oxytocin can heighten negative emotions, particularly when it comes to social exclusion or perceived betrayal. Research has shown that oxytocin can intensify feelings of jealousy or defensiveness when people feel their relationships are threatened. In an experiment where participants were given oxytocin and then exposed to images of their romantic partner interacting with someone else, they showed increased feelings of jealousy and possessiveness. This suggests that while oxytocin can enhance trust and attachment, it can also amplify protective or territorial instincts. The key takeaway from these studies is that oxytocin does not automatically create trust, nor does it act in a vacuum. Trust is built through ongoing, reciprocal social interactions. Oxytocin facilitates this process by making people more attuned to positive social cues, reinforcing connections and reducing fear and anxiety in safe, familiar relationships. It plays a role in strengthening the emotional bonds between people, but it does not bypass the complexities of human connection. Trust requires time, consistency, and mutual vulnerability, factors that oxytocin supports but does not independently create. The psychological function of oxytocin also extends beyond interpersonal relationships. It plays a role in the broader social fabric. In communities or societies, oxytocin may help reinforce cooperative behavior, encouraging people to work together for mutual benefit. In one study, participants who received oxytocin showed increased cooperation in group tasks, demonstrating the hormone's role in promoting social bonding at a larger scale. Oxytocin's effect on cooperation and pro-social behavior has led some to consider its potential applications in therapy and conflict resolution. For example, individuals with social anxiety or those recovering from trauma may benefit from treatments that help modulate oxytocin systems, fostering a greater sense of safety and social connection. However, much like its effects on trust, these interventions are not a panacea. They must be combined with other therapeutic approaches to be effective. The use of oxytocin as a therapeutic tool also raises ethical questions. If oxytocin can be used to enhance trust or empathy, should it be administered in contexts where it could alter relationships artificially? Could it be used to manipulate emotions or influence behavior without consent? As researchers continue to explore the possibilities of oxytocin in clinical settings, these questions will become increasingly important. Despite the challenges and limitations of studying oxytocin's role in trust, its involvement in human social behavior is undeniable. Oxytocin shapes how we perceive others, how we respond to social cues, and how we form lasting emotional bonds. It enhances trust when that trust is already present, amplifies empathy, and helps to reinforce social connections. But oxytocin is not a magic key that opens the door to instant trust. It is part of a larger system that includes cognition, past experiences, and social context. As we explore the role of oxytocin in human relationships, it's essential to keep in mind that while this hormone plays a pivotal role in creating the conditions for trust and bonding, it works in concert with other psychological and social factors. Love, connection, and trust are not simply chemicals in the brain. They are lived experiences shaped by interaction, communication, and shared vulnerability. Oxytocin facilitates these experiences, but it cannot replace the deeper, richer process of building trust that unfolds over time. Oxytocin is often described as a feel-good hormone, a chemical messenger that facilitates bonding, trust, and emotional connection. For many, this reputation has led to the belief that oxytocin is always beneficial, always supportive of positive social behavior. Yet as with many chemicals in the brain, oxytocin's effects are not straightforward, and they can sometimes behave in unexpected or even counterproductive ways. Understanding when and why oxytocin doesn't behave as expected helps us grasp the complexity of its role in social dynamics. While oxytocin is crucial for forming social bonds, enhancing trust, and encouraging cooperation, its effects are not always benign. In fact, Oxytocin can sometimes amplify negative behaviours, especially in social contexts that involve group dynamics, in-group favouritism, or social exclusion. This dual nature of oxytocin complicates its image as the love hormone. Rather than merely promoting positive interactions, it can also reinforce divisiveness or exacerbate certain emotions in specific situations. In-group bias and out-group hostility. One of the most striking examples of oxytocin's unexpected behaviour is its tendency to increase in-group favouritism and out-group hostility. Research has shown that when oxytocin levels rise, people are more likely to show trust and generosity toward individuals they perceive as part of their social group. This is a well-documented effect, but it has a darker side. Oxytocin can also heighten suspicion, prejudice, and negative feelings toward those outside the group. In studies where participants were given intranasal oxytocin, they demonstrated greater loyalty and kindness toward their in-group members, but they were also more likely to express negative views toward out-group members. The hormone seemed to intensify the perception of difference between us and them, reinforcing social boundaries rather than promoting universal compassion. This effect may have evolutionary roots. Oxytocin's role in promoting attachment and bonding within social groups is essential for the survival and stability of human communities. In ancestral environments, close-knit groups were more successful in cooperative tasks such as hunting, defense, and child rearing. By strengthening bonds within the group, oxytocin helped ensure that individuals worked together to protect shared resources. However, when released in a context that promotes exclusivity, oxytocin can also heighten distrust and suspicion of outsiders, an effect that was beneficial for group cohesion, but less so in modern multicultural societies. Jealousy and possessiveness. Another scenario where oxytocin doesn't behave in a straightforward, positive manner is in the context of romantic relationships. While oxytocin supports long-term bonding and attachment, it can also fuel jealousy, possessiveness, and territorial behaviours, particularly when a relationship feels threatened. In romantic partnerships, oxytocin has been linked to heightened feelings of jealousy when one partner perceives a threat from a third party. Research in both humans and animals has shown that oxytocin levels rise in response to infidelity or the threat of infidelity. In experiments where individuals were exposed to images of their romantic partner interacting with someone else, oxytocin administration increased feelings of jealousy and possessiveness. This is not necessarily a negative outcome. Jealousy can be seen as an adaptive emotion that protects relationships and ensures mating opportunities. But when excessive, it can undermine trust and cause emotional distress. In one study, couples who were given oxytocin before a stressful interaction demonstrated more positive communication during conflict resolution. However, when similar couples were exposed to a perceived threat, such as their partner interacting with an attractive stranger, oxytocin amplified feelings of jealousy and increased protective or defensive behaviours. Oxytocin, in this case, did not reduce anxiety or enhance communication. Instead, it increased emotional reactivity and territorial instincts. This suggests that while oxytocin promotes feelings of safety and connection in secure relationships, it also heightens emotional responses to perceived threats. In situations where insecurity or jealousy is already present, oxytocin can worsen those feelings, exacerbating the very issues it is supposed to help resolve. Social exclusion and oxytocin's dark side. While oxytocin is commonly associated with social inclusion, positive group behaviour, and social bonding, its effects can flip in situations involving social exclusion. Oxytocin's role in enhancing in-group bonding means that when individuals are excluded from a group, they may experience a greater sense of emotional pain. In fact, oxytocin is involved in the brain's processing of social rejection, not just social connection. Studies examining the neural correlates of social exclusion have found that oxytocin is released in response to feelings of rejection, but this release may amplify the emotional pain associated with being excluded. In social exclusion experiments, participants who were ostracized by a group of peers showed increased oxytocin levels. Rather than buffering the pain of exclusion, oxytocin seemed to amplify the emotional distress associated with being rejected. This paradoxical effect of oxytocin underscores its complexity. In one study, participants who were given oxytocin after experiencing social exclusion reported more emotional distress than those who received a placebo. Instead of mitigating the negative impact of rejection, oxytocin appeared to heighten the sense of emotional vulnerability. This suggests that oxytocin's effects depend heavily on the social context, and that it can either support or hinder emotional regulation depending on whether the environment is one of inclusion or exclusion. Over-reliance on oxytocin for connection. Another potential downside of oxytocin's effects is its influence on how people approach emotional connection. In some contexts, individuals may become overly reliant on the hormone to facilitate trust or emotional closeness. While oxytocin can help foster positive social behaviors, it is not a substitute for the work required to build and maintain deep, meaningful relationships. When oxytocin is administered externally, such as through nasal sprays or other forms of supplementation, it can create short-term feelings of closeness or relaxation. However, these feelings are fleeting. The absence of a reciprocal emotional connection can render these effects shallow. Oxytocin's ability to boost feelings of attachment requires context. Without real, meaningful social interactions, its effects can be temporary and may not lead to lasting emotional bonds. Furthermore, the over-reliance on oxytocin to stimulate positive social interactions can have unintended consequences. Individuals may expect oxytocin's effects to do the work of emotional labor, reducing the motivation to engage in the effort required to cultivate trust, manage conflict, and navigate the complexities of human relationships. Oxytocin and aggression. While oxytocin is most often associated with prosocial behaviors, there is evidence to suggest that it can also influence aggression, especially in competitive contexts. Oxytocin has been linked to increased aggression toward individuals perceived as threats or competitors. In animal studies, oxytocin administration has been shown to enhance territorial behaviors, making animals more likely to engage in defensive or aggressive acts in the presence of intruders. In humans, oxytocin's effects on aggression are not as clearly defined, but research has shown that it can enhance aggressive responses in certain situations. For example, oxytocin administration has been shown to increase aggressive behavior in competitive social contexts, where individuals feel their position is being challenged. In one study, participants who received oxytocin were more likely to act aggressively in response to perceived insults or challenges. This suggests that oxytocin may act as a modulator, enhancing aggression when it's triggered by perceived threats, but it does not universally promote calm or prosocial behavior. Conclusion. Oxytocin is a powerful molecule influencing many aspects of human behavior, from trust and bonding to emotional regulation and stress response. However, its effects are not always positive, nor are they universally beneficial. In certain contexts, oxytocin can amplify jealousy, possessiveness, in-group bias, and aggression. It can also heighten the emotional distress caused by social exclusion, and it is not a shortcut for building genuine, lasting relationships. The key to understanding oxytocin's behavior lies in its context. It enhances connection and trust when those bonds are already present, or when the social environment is safe and supportive. But it can also magnify negative emotions or social divisions when the environment is stressful, competitive, or marked by exclusion. Oxytocin's role in shaping human behavior is not as simple as trust in a bottle. It is a powerful, context-dependent tool that interacts with emotional and social dynamics in complex ways. Understanding when and why oxytocin doesn't behave the way we expect it to helps us navigate the delicate balance between connection and division, love and conflict in human relationships. Attachment theory, originally developed by psychologist John Bowlby, posits that the bonds we form with our primary caregivers during childhood shape the way we approach relationships throughout life. Attachment styles, secure, anxious, and avoidant, describe how individuals behave in close relationships, and they are often formed in response to early experiences with caregivers. However, what's becoming increasingly clear is that these attachment styles are not just psychological constructs. They also have a strong biological foundation. Neurochemicals, particularly oxytocin, play a critical role in how attachment styles develop and are expressed later in life. These chemical signatures influence the way we form bonds, manage emotional intimacy, and navigate social connections. Oxytocin, often called the bonding hormone, has been linked to the creation and maintenance of attachment throughout life. Its effects on trust, social connection, and emotional regulation are well documented. However, recent research suggests that the way we respond to oxytocin and the intensity of its effects may differ depending on our attachment style. Understanding this relationship can shed light on how early experiences shape our emotional and physiological responses to others, and why some people find it easier to form close, trusting relationships while others struggle. The secure attachment style. Secure attachment is often considered the ideal attachment style. Individuals with a secure attachment style generally feel comfortable with intimacy, trust their partners, and are able to communicate openly about their emotions. They are also able to manage conflict effectively and are generally resilient in the face of stress or adversity. Securely attached individuals tend to have positive views of themselves and others, and they typically have satisfying long-term relationships. Biologically, secure individuals tend to have more balanced and stable oxytocin responses. When oxytocin is released in response to social interaction, they experience its calming and bonding effects in a predictable, beneficial way. This consistent oxytocin release reinforces the sense of safety and connection, which in turn strengthens the attachment bond. Research has shown that securely attached individuals tend to exhibit a strong social orientation, meaning they are more likely to seek and enjoy positive social interactions. They show greater sensitivity to positive social cues and are generally more attuned to the emotional states of others. Their oxytocin systems seem well calibrated to social engagement. When they are in secure, supportive relationships, oxytocin helps regulate their stress levels and enhances their sense of emotional connection. One key aspect of secure attachment is the ability to balance independence with closeness. Securely attached individuals are not overly reliant on others for emotional regulation, but they still seek and appreciate close, supportive relationships. Oxytocin plays a role in this balance, helping individuals feel connected without becoming overly dependent on others for emotional validation. The anxious attachment style. Individuals with an anxious attachment style often have intense emotional responses to relationship dynamics. They tend to worry about abandonment, seek constant reassurance, and may become overly dependent on their partner for validation. Their emotional experience in relationships is often characterized by high levels of anxiety, fear of rejection, and a tendency to cling to their partner in times of stress. These individuals often have a negative view of themselves and are hypersensitive to perceived threats to the relationship. For people with an anxious attachment style, oxytocin may not behave in the same predictable, stabilizing way. While oxytocin is still released in response to social bonding, the intensity of the hormone's effect can be exaggerated. When anxiously attached individuals form connections, they may experience a stronger physiological and emotional reaction than those with a secure attachment style. This heightened response can result in increased feelings of dependence on their partner and a greater need for constant emotional reassurance. In some cases, the release of oxytocin in response to social interaction can be dysregulated. Instead of producing a calming effect, it may amplify anxiety and emotional instability, particularly in response to perceived relationship threats. This could explain why individuals with anxious attachment styles are often more prone to jealousy, preoccupation with their partner's actions, and heightened sensitivity to relationship dynamics. Interestingly, studies have shown that anxious individuals may have an overactive hypothalamic pituitary adrenal (HPA) axis, which governs the stress response. When oxytocin is released in these individuals, it may not counterbalance their stress response in the same way it does for securely attached individuals. Instead, it can heighten emotional reactivity, leading to a cycle of attachment-related anxiety. This pattern suggests that while oxytocin can be beneficial in promoting bonding and reducing stress, its effects are less predictable in people with anxious attachment. The biological underpinnings of this attachment style involve a complex interaction between hormonal responses, emotional regulation, and learned patterns of seeking connection. The avoidant attachment style. Avoidant attachment is characterized by emotional distance and a reluctance to depend on others. Individuals with an avoidant attachment style typically value independence and self-sufficiency over emotional closeness. They may have difficulty expressing their emotions, and they often downplay the importance of relationships or intimacy. This style can develop when caregivers are consistently unavailable, unresponsive, or dismissive of the child's emotional needs. Oxytocin's effects in individuals with an avoidant attachment style are often weaker or less pronounced. These individuals may show less of the typical oxytocin-mediated bonding response during positive social interactions. While oxytocin is still involved in their emotional processing, its release may be less frequent or less effective in facilitating connection. The physiological benefits of oxytocin, such as stress reduction and emotional regulation, may not be as readily accessible to avoidantly attached individuals, as they tend to resist closeness and emotional vulnerability. When oxytocin is released in response to intimacy or social bonding, avoidantly attached individuals may experience discomfort rather than the typical feelings of warmth and connection. Their brains may interpret the hormone's effects as threatening to their emotional autonomy, and they may actively suppress the emotional response that oxytocin promotes. This can contribute to their tendency to downplay the importance of emotional closeness and avoid seeking support in times of distress. This lack of responsiveness to oxytocin may be related to differences in receptor sensitivity. People with avoidant attachment styles may have fewer oxytocin receptors or may not respond as strongly to the hormone's effects. Additionally, avoidantly attached individuals may have a heightened cortisol response to social interaction, particularly in situations that involve intimacy or vulnerability. This could create a physiological barrier that prevents oxytocin from functioning in its typical, stabilizing role. In contrast to anxious individuals who crave closeness and reassurance, avoidantly attached individuals tend to withdraw when faced with emotional demands. They may reject or shut down when they perceive emotional closeness as overwhelming. This emotional withdrawal can prevent oxytocin from functioning as a bonding agent and reinforce the distance they feel in relationships. The complexity of attachment and neurochemistry. The biological signatures of attachment styles underscore the complexity of human relationships. Oxytocin is a powerful modulator of social behavior, but its effects are not one-size-fits-all. It interacts with other hormones, such as cortisol, and is influenced by individual differences in receptor distribution and sensitivity. Attachment styles shaped by early experiences and environmental factors determine how oxytocin functions within the body and brain. The varying effects of oxytocin across attachment styles also highlight the importance of early attachment experiences in shaping our neurobiological responses to intimacy. A person's attachment style is not just a psychological construct, it has a tangible biological basis. Early caregiving experiences can influence the development of oxytocin receptors, the sensitivity of the HPA axis, and the way the brain processes emotional cues. These biological patterns, in turn, affect how individuals form and maintain relationships throughout their lives. Understanding these chemical signatures provides insight into why certain individuals may struggle with trust, emotional regulation, and intimacy, while others seem to form stable, secure relationships more easily. It also suggests that attachment patterns are not fixed or immutable. With therapy and positive relationship experiences, it is possible to recalibrate the neurochemical systems that influence attachment making it possible for individuals to develop healthier relationships and increase their capacity for emotional connection. In summary, oxytocin is a key player in the biology of attachment, but its effects are shaped by an individual's attachment style. Securely attached individuals experience oxytocin's calming and bonding effects in a stable and predictable manner. In contrast, individuals with anxious or avoidant attachment styles may experience oxytocin differently, with varying levels of emotional reactivity, stress, and interpersonal distance. By exploring the intersection of attachment theory and neurochemistry, we gain a deeper understanding of how our earliest relationships shape not only our emotional experiences, but also the biological systems that regulate them. Loneliness and isolation are often thought of as emotional experiences, feelings of sadness, emptiness, or longing for connection. However, they are not just psychological states, they are deeply embedded in our biology. When we experience loneliness or social isolation, our brains react in ways that are similar to physical pain, highlighting how central social connection is to our survival and well-being. These emotional states trigger complex neural and hormonal responses that affect everything from stress levels to immune function, reinforcing the idea that loneliness is not simply a feeling, but a physiological condition. One of the key factors in how loneliness impacts the brain is the disruption of social bonding systems, particularly those involving oxytocin, which plays a significant role in fostering connections between individuals. The absence of social contact or supportive relationships disrupts the normal function of these systems and leaves the brain vulnerable to negative outcomes both emotionally and physically. The brain's response to social isolation. Humans are social creatures by nature, and our brains are wired to thrive in the company of others. This wiring is linked to evolutionary survival. For early humans, being part of a group was crucial for finding food, protecting against predators, and ensuring the survival of offspring. In modern times, the need for social connection remains deeply ingrained in our biology, despite the shift away from survival-focused interactions. When people experience loneliness or isolation, the brain responds as though it is facing a threat. The absence of social connection triggers a cascade of neurobiological changes, many of which are similar to the body's response to physical pain or stress. The brain's default mode network, which is involved in self-reflection and processing internal states, becomes more active in isolated individuals. This can lead to increased rumination, repeatedly thinking about one's feelings of loneliness or isolation, which in turn exacerbates emotional distress. One of the key brain regions involved in this process is the anterior cingulate cortex, ACC. The ACC is involved in processing both physical pain and social pain, including the emotional pain that comes from feeling excluded or disconnected. Studies have shown that the ACC activates in response to social rejection or exclusion in the same way it does when individuals experience physical pain. This suggests that the brain does not differentiate between physical and social pain, reinforcing the idea that loneliness and social isolation are felt in the body as much as they are in the mind. In addition to the ACC, the amygdala, a region of the brain involved in processing fear and threat, becomes more active when a person feels isolated. The amygdala is central to the brain's response to perceived threats, and when it perceives a lack of social support, it triggers a stress response. This can lead to heightened anxiety, hypervigilance, and emotional distress. Over time, the chronic activation of the amygdala due to prolonged loneliness or social isolation can increase susceptibility to anxiety disorders, depression, and other mental health conditions. The role of oxytocin in loneliness. Oxytocin, often called the bonding hormone, plays a central role in social connection. It is released during positive social interactions, such as hugging, touching, or spending time with loved ones, and promotes feelings of trust, safety, and emotional connection. When oxytocin is released, it reduces the body's stress response and enhances feelings of well-being and attachment. It also acts to regulate the autonomic nervous system, helping to reduce heart rate and blood pressure, making the body feel calmer and more relaxed. However, in the absence of social connection, oxytocin levels can drop, leading to a cascade of negative effects on the body and brain. Chronic loneliness has been shown to decrease oxytocin receptor availability in the brain, which may exacerbate feelings of social disconnection and make it harder to experience the emotional and physiological benefits of connection. Moreover, when oxytocin levels are low, the body becomes more vulnerable to stress. Without the calming influence of oxytocin, the stress hormone cortisol rises. Prolonged elevated cortisol levels due to social isolation can impair immune function, increase inflammation, and contribute to chronic health issues like heart disease, hypertension, and gastrointestinal problems. This hormonal imbalance illustrates how loneliness and isolation are not just mental health concerns, but are linked to physical health as well. The social pain of loneliness activates the body's stress systems, making it harder for the body to return to a state of calm and repair, which can lead to long-term negative health consequences. Loneliness and the immune system. The impact of loneliness on the immune system is one of the most significant aspects of how isolation affects health. Chronic loneliness has been linked to increased levels of inflammation in the body. This is partly because of the increased release of cortisol, which in turn triggers the release of pro-inflammatory cytokines. These cytokines are involved in the body's immune response to injury or infection, but when they are chronically elevated due to prolonged stress, they can contribute to the development of various chronic diseases. In studies examining the effects of social isolation on the immune system, researchers have found that individuals who are socially isolated have a weakened immune response to vaccines and other infections. For example, one study showed that lonely individuals produced fewer antibodies in response to a flu vaccine compared to their more socially connected counterparts. This reduced immune efficiency makes isolated individuals more susceptible to illness and infections, demonstrating the far-reaching effects of loneliness on the body's ability to protect itself. Social connection as a buffer to stress. While loneliness exacerbates the stress response, social connection has the opposite effect. Having supportive social relationships, whether they are romantic, familial, or platonic, acts as a powerful buffer to stress. People with strong social ties tend to have lower levels of cortisol and are more resilient in the face of stress. In fact, the presence of close social connections has been shown to decrease the physiological effects of stress and promote recovery from illness. This buffering effect is largely mediated by oxytocin. When people are socially connected, oxytocin levels increase, promoting relaxation, reducing the stress response, and enhancing overall well-being. Social support helps regulate the body's response to stress, making it easier to manage life's challenges and reducing the risk of developing stress-related health issues. The importance of social connection for health is evident across the lifespan. Children who grow up with secure attachments to caregivers tend to have better emotional regulation, lower levels of anxiety, and better physical health outcomes. Adults with strong social networks report higher levels of life satisfaction, better cardiovascular health, and lower rates of depression. The positive effects of social connection even extend into old age, where individuals with meaningful social relationships tend to live longer and experience better cognitive and physical health. The brain's capacity for change, neuroplasticity, and recovery. Although loneliness and isolation have profound effects on the brain is also capable of change. Neuroplasticity, the brain's ability to reorganize itself by forming new neural connections, allows for recovery from the damaging effects of social isolation. Just as early experiences with caregivers shape attachment systems, later experiences with social connection can reshape the brain's response to loneliness. This means that even individuals who have experienced long periods of isolation can still re-engage socially and experience the benefits of connection. Positive social interactions can stimulate oxytocin release and help restore balance to the hormonal and neural systems that were disrupted by loneliness. Social support, therapy, and community involvement can all play a role in this recovery process, helping individuals rebuild their social networks and re-establish healthy emotional and physiological responses to connection. In clinical settings, interventions designed to reduce loneliness and encourage social engagement have shown positive effects. Group therapy, social activities, and community-building programs can increase oxytocin levels, reduce cortisol, and improve overall well-being. These interventions are particularly important for individuals who are at risk of chronic isolation, such as the elderly or those with mental health disorders. Conclusion: loneliness and social isolation have a profound impact on the brain and body, leading to increased stress, weakened immune function, and heightened emotional distress. The disruption of oxytocin's calming and bonding effects is central to this process, leaving individuals vulnerable to the negative effects of isolation. However, the brain's capacity for change offers hope. Through social connection, oxytocin levels can be restored, stress responses can be regulated, and the physical and emotional effects of loneliness can be mitigated. Recognizing the biological underpinnings of loneliness emphasizes the importance of building and maintaining social connections, not just for emotional well-being, but for physical health as well. Social isolation is not just a mental health issue. It is a physiological one that can be addressed by fostering community, support, and connection. Romantic love is often depicted in popular culture as a singular, intense, and passionate experience, something that is all-consuming in its early stages and gradually fades as time passes. But the reality of romantic love is far more complex, and the experience of love evolves as relationships progress. While the initial stages of romantic love are characterized by intense attraction and infatuation, long-term relationships tend to shift from this heightened emotional state into something more stable, grounded, and emotionally rewarding. The key to understanding this transformation lies in the hormonal and neural changes that accompany the evolution of love over time. The early stages: passion and infatuation. In the beginning, romantic love is often marked by infatuation, a state of intense attraction that feels exciting, unpredictable, and all-consuming. During this phase, dopamine and norepinephrine surge in the brain, creating feelings of excitement, energy, and heightened alertness. Dopamine, the reward neurotransmitter, is particularly involved in the pursuit of pleasure and motivation. It fuels the desire to seek out the person you're attracted to obsess over small details, and to experience the thrill of anticipation. This initial rush is often referred to as the honeymoon phase of a relationship, during which everything feels new, exciting, and full of possibility. At the same time, norepinephrine, which is associated with arousal and energy, heightens physical responses such as an increased heart rate, sweaty palms, and nervous excitement. These sensations are often described as the butterflies that come with falling in love. The combination of dopamine and norepinephrine creates an addictive quality to infatuation, as the brain gets used to the pleasure of new experiences and interactions with a romantic partner. While these chemical signals foster intense attraction, they are not the only players involved. Cortisol, the stress hormone, can also rise during the early stages of romantic love, adding a layer of tension or excitement to the experience. The uncertainty of whether the attraction is mutual, the anticipation of a first kiss, or the anxiety of not knowing where the relationship is headed can all increase cortisol levels. In this way, early love is a cocktail of excitement, stress, and reward. The transition to attachment, oxytocin, and stability. As the relationship progresses, the intense feelings of infatuation begin to stabilize. The passion-driven rush of dopamine and norepinephrine starts to ebb, and the experience of love becomes more grounded in emotional connection, trust, and security. During this phase, oxytocin plays a central role. Oxytocin, often called the bonding hormone, is released during moments of physical affection, such as hugging, kissing, and sexual intimacy. It is also released when partners engage in emotionally intimate behaviours, such as sharing vulnerable thoughts or providing comfort during stressful times. Oxytocin's role in attachment is crucial. It helps to transform the initial excitement of infatuation into a deeper, more lasting bond. The hormone promotes feelings of safety, trust, and emotional regulation. When oxytocin is released, it dampens the brain's fear responses and enhances the brain's reward centers, creating a sense of calm and contentment when with a partner. In this way, oxytocin reinforces the positive associations we develop with our romantic partner over time, shifting the experience of love from excitement to security. As oxytocin strengthens the bond, the relationship becomes more about shared experiences, mutual support, and emotional intimacy. The intensity of early passion may fade, but it is replaced by a deeper connection built on trust and comfort. The brain begins to prioritize the presence of the partner as a source of emotional stability and safety, rather than as a new, exciting stimulus. This shift marks the transition from the passion of the initial phase to the more enduring love that is characteristic of long-term relationships. The role of vasopressin in long-term bonding. In addition to oxytocin, vasopressin, a hormone structurally similar to oxytocin, plays a key role in long-term bonding, particularly in men. Vasopressin is involved in social behavior bonding and territoriality. It has been shown to reinforce monogamous behaviors in certain species, including humans. In the early stages of a relationship, vasopressin may contribute to the desire to spend time with and protect a partner. Over time, it supports the commitment and loyalty that are central to long-term relationships. Vasopressin also works in conjunction with oxytocin to reinforce attachment behaviors, but its role tends to be more focused on the protective and caregiving aspects of the relationship. While oxytocin enhances emotional closeness and reduces anxiety, vasopressin plays a role in promoting behaviors that ensure the long-term stability of the bond. It may also be involved in the formation of social memory, helping individuals remember and recognize their partner as a key source of support. In this way, vasopressin complements oxytocin in reinforcing long-term attachment. Together, they help maintain the bond that forms between partners, allowing them to navigate the challenges of a shared life. The enduring nature of love, attachment, and shared history. As a relationship matures, the chemical signatures of early romantic love continue to shape how partners relate to one another. While the dopamine-driven excitement of infatuation may fade, the role of oxytocin and vasopressin becomes even more important. Over time, relationships become less about the emotional highs of early passion and more about the quiet, steady support that partners provide each other. One of the key factors in maintaining long-term love is the accumulation of shared experiences. Every moment of emotional support, every challenge overcome together, and every act of kindness strengthens the neural pathways that associate the partner with feelings of security, safety, and trust. These positive experiences create a narrative of the relationship in the brain, reinforcing the emotional connection over time. Additionally, the memories that are encoded during moments of closeness are influenced by oxytocin. Oxytocin strengthens the emotional tone of experiences, making them more vivid and more likely to be recalled. The shared history between partners becomes a key part of the bond, as each positive experience serves as a building block for future connection. This process reinforces the sense of attachment, helping partners maintain their emotional bond even as the intensity of early love fades. The hormone's role in memory also helps explain why long-term relationships can feel so rewarding. The brain's association of the partner with emotional safety and comfort grows stronger over time, making the relationship more resilient to stressors. Partners who have developed a deep emotional connection are better able to weather challenges together because their bond is rooted in a long history of positive, supportive interactions, maintaining passion in long-term relationships. While the initial passion of romantic love tends to evolve into a more stable attachment, it's important to note that long-term relationships don't have to lose their emotional intimacy or physical affection. In fact, research suggests that relationships that maintain passion and physical closeness over time tend to have healthier outcomes. Even in established relationships, regular physical affection, whether through touching, kissing, or sexual intimacy, can continue to stimulate oxytocin release and keep the emotional bond strong. Couples who engage in novel activities together, maintain physical intimacy, and continue to invest in their emotional connection are more likely to maintain a satisfying romantic relationship over the years. This reinforces the idea that love in long-term relationships requires effort and attention. While the initial intensity of passion may fade, it can be sustained through intentional acts of affection, communication, and emotional support. Conclusion: romantic love is not a static experience. It evolves over time. The early stages of love are marked by intense passion, excitement, and attraction, fueled by dopamine and norepinephrine. As the relationship matures, love shifts toward attachment and security, driven by the release of oxytocin and vasopressin. These hormones promote emotional closeness, trust, and stability, forming the foundation for long-term bonds. While the early infatuation may fade, the love that develops over time can be deeper, richer, and more enduring. Long-term relationships rely on shared history, emotional regulation, and mutual support. The neurochemicals involved in love, particularly oxytocin, help maintain the bond, making it resilient to stress and challenges. Understanding the hormonal and neural basis of romantic love over time underscores the complexity of human relationships. Love is not just about initial attraction. It is about building a lasting, supportive connection that evolves as both individuals grow. This process, which involves not only chemicals but also shared experiences and emotional investments, helps sustain love and attachment through the ups and downs of life. In today's digital age, social media and screens have become central to the way people connect with each other. Platforms like Facebook, Instagram, Twitter, and TikTok have revolutionized the way we maintain relationships, build communities, and share experiences. While these technologies offer unprecedented opportunities for social interaction, they also raise important questions about how virtual connections influence our emotional well-being, social bonding, and the neurochemistry of attachment. Social media is often touted as a way to stay connected, particularly for individuals who live far from family or friends, or who have limited opportunities for face-to-face interaction. However, the nature of these interactions is different from in-person relationships, and it may have significant effects on how we experience connection and loneliness, as well as how our brains process and regulate social bonding. The evolution of social connection in the digital age. Human beings have evolved in environments where face-to-face interaction was the primary means of communication. The brain has developed specialized systems for processing social information, recognizing faces, interpreting emotions, and detecting social cues. These systems were honed through direct physical interactions with others. Our attachment systems, those that regulate bonding and connection, are deeply rooted in these in-person experiences. However, in the digital age, screens have replaced many of these in-person interactions. The question arises, how does this shift impact the neurochemistry of connection? Are virtual connections as effective in promoting bonding as face-to-face encounters? And if not, what implications does this have for our emotional health and relationships? The role of oxytocin in virtual connections. Oxytocin, the hormone most commonly associated with bonding, trust, and emotional connection, is released during physical touch, shared eye contact, and intimate social exchanges. It plays a central role in reinforcing the bonds between individuals, whether in parent-child relationships, friendships, or romantic partnerships. The hormone fosters feelings of safety, trust, and emotional closeness. In face-to-face interactions, oxytocin is released naturally during physical touch, such as hugging or holding hands, and even through social cues like eye contact or a warm smile. These interactions engage the brain's attachment system and promote feelings of emotional connection. But does oxytocin behave the same way when interactions occur through a screen? Research suggests that while social media interactions can lead to some degree of oxytocin release, it is much less potent than in-person bonding. Social media platforms provide many opportunities for shared experiences, comments, likes, reactions, and messages that mimic some of the cues that promote attachment in face-to-face interactions. However, the lack of physical proximity and touch means that the neurobiological effects of these interactions are often weaker and more fleeting. Interestingly, oxytocin released through virtual connections may be less about the content of the interaction, a like, comment, or share, and more about the feeling of being seen or acknowledged. For example, receiving a like on a post or a positive comment can activate the brain's reward system, providing a small boost of pleasure and connection. However, these interactions lack the depth and richness of in-person bonding, which includes physical touch, the ability to read subtle body language, and the emotional resonance that comes with shared experiences. The illusion of connection and the loneliness paradox. While social media can create the illusion of connection, it can also lead to feelings of isolation. Studies have shown that despite the fact that people spend hours each day on social media platforms, they report feeling lonelier than ever. This paradox, feeling more isolated despite being constantly connected, may be linked to the nature of digital interactions themselves. One issue is that social media often encourages shallow interactions. Likes, comments, and shares are quick, often superficial responses that lack the emotional depth and intimacy of face-to-face conversations. These interactions may provide a brief sense of validation, but they do not lead to the kind of deep emotional connection that oxytocin facilitates in more personal settings. Furthermore, the curated nature of social media, where users selectively present idealized versions of their lives, can exacerbate feelings of loneliness. When people compare their own experiences to the polished highlight reel lives of others, they may feel that their own lives are lacking or that they are disconnected from others. This sense of comparison can increase feelings of isolation and contribute to anxiety, depression, and dissatisfaction with one's social life. When individuals rely on digital interactions for emotional fulfillment, they may experience a disconnect between the positive reinforcement provided by online engagement and the deep emotional satisfaction that comes from real-life relationships. Over time, this reliance on virtual validation can weaken the brain's attachment systems, making it more difficult to form lasting emotional connections. Social media and the dopamine-driven cycle of reward. While oxytocin plays a role in creating lasting emotional bonds, another neurochemical, dopamine, is closely linked to the addictive aspects of social media. Dopamine is the brain's reward neurotransmitter associated with motivation, pleasure, and reinforcement. When we receive positive feedback on social media, whether it's a new notification, a comment, or a like, dopamine is released, creating a feeling of pleasure and reinforcing the behavior that led to the reward. This dopamine-driven feedback loop can make social media highly addictive. The anticipation of a notification or a like triggers dopamine release, which motivates individuals to engage more frequently with their platforms. This cycle can lead to excessive use of social media, often at the expense of in-person social interactions. While dopamine provides a temporary pleasure boost, it does not provide the long-lasting emotional satisfaction that oxytocin-mediated bonding offers. The result is that individuals may feel momentarily gratified by virtual interaction, but remain emotionally unfulfilled, leading to a deeper sense of loneliness and dissatisfaction. Screens and the impact on empathy and emotional regulation. In addition to its impact on oxytocin and dopamine, the shift to virtual connection also affects other key aspects of emotional regulation and empathy. In face-to-face interactions, we rely on a wide range of non-verbal cues, facial expressions, body language, tone of voice, to gauge the emotional states of others and respond appropriately. These cues are crucial for building empathy and emotional understanding. However, digital communication often lacks these cues. Text-based interactions, in particular, are stripped of the nuances of facial expressions and body language, making it harder to fully understand the emotional context of the message. This can result in miscommunications, misunderstandings, and emotional disconnection. Furthermore, the overuse of digital communication can impair our ability to read and respond to social cues in person, leading to a decline in overall social intelligence and empathy. Research has shown that people who engage heavily with screens, whether through social media, video games, or other digital platforms, may have reduced activity in areas of the brain associated with empathy, such as the medial prefrontal cortex. This suggests that constant digital interaction may be weakening our ability to engage emotionally with others and respond to their needs in meaningful ways. The role of screens in building community. Despite the challenges posed by virtual interactions, it's important to recognize that screens and social media can still play a positive role in building and maintaining relationships. For people who are geographically distant from family and friends, digital platforms offer a way to stay connected, share experiences, and provide support. Video calls, for example, allow people to see each other's faces and hear each other's voices, which can enhance the sense of connection compared to text-based communication alone. Social media platforms can also create spaces for marginalized groups to find community, share experiences, and offer support. Online communities centered around shared interests, identities, or challenges can provide a sense of belonging that might be difficult to find in physical spaces. For many, these communities provide a crucial support network, particularly for those who may not have access to these resources locally. Conclusion: the balancing act of connection. In the digital age, screens and social media have fundamentally altered the way we connect with one another. While these platforms provide new opportunities for communication and interaction, they also present challenges for emotional well-being and neurochemistry. While social media can lead to small bursts of pleasure and connection, it often falls short in providing the deep emotional bonds that are necessary for long-term happiness. The rise of digital interaction has changed the way oxytocin and dopamine influence our relationships, reinforcing the need for a careful balance between virtual and in-person connection. In the end, social media and screens are not inherently harmful, but they require mindful usage. Face-to-face interactions that include physical touch, empathy, and vulnerability remain the gold standard for building lasting, meaningful connections. It is important to recognize that virtual interactions can complement these bonds, but should not replace the kind of social engagement that allows oxytocin and emotional connection to flourish. By fostering deeper, more authentic connections, both online and offline, we can ensure that technology enhances our relationships rather than diminishes them. When we speak about love, it often feels like something beyond biology, something mysterious, magical, and deeply human. We talk about love in poetic terms, describing it as a force that transcends logic, a feeling that goes beyond chemistry. Yet, at its core, love is also profoundly tied to chemistry, neurochemicals, hormones, and brain circuits that influence how we feel, think, and behave in relationships. So, the question arises, is love just chemistry? To answer this, it's important to first understand that love, in its many forms, is a complex and multifaceted experience. It involves not only the biological processes that occur in the brain and body, but also psychological, social, and cultural factors. While love cannot be reduced to a single chemical reaction, there's no denying that our brain's chemistry plays a foundational role in how we experience connection, attraction, and attachment. The chemistry of attraction, the initial spark. At the onset of romantic love, the brain experiences an intense rush of neurochemicals. This is often called the honeymoon phase. characterized by heightened emotional and physical responses to the presence of a partner. The brain's reward system goes into overdrive. Dopamine, the neurotransmitter associated with pleasure and reward, plays a significant role in this early stage. When we are in love, dopamine is released and we experience feelings of euphoria, motivation, and desire to pursue the object of our affection. This is why falling in love often feels so exciting and overwhelming. It's a chemical high. In addition to dopamine, norepinephrine, which is involved in the body's stress response, also spikes during early infatuation. This hormone increases heart rate, blood pressure, and the feeling of physical arousal. The combined effect of dopamine and norepinephrine explains why we can feel so charged when we're in the early stages of a relationship. Our brains are essentially rewarding us for being close to the person we desire, and our bodies react accordingly with a rush of sensations that accompany the initial stages of attraction. However, this initial stage is just the beginning. While the early stages of love are largely driven by these rewarding neurochemicals, the experience of love itself becomes more complex and deepened over time, as we move from the infatuation stage to long-term attachment. The evolution of attachment, oxytocin and vasopressin. As romantic love progresses, the brain shifts from the excitement of infatuation to the stability of long-term attachment. This transition is driven by oxytocin and vasopressin, two hormones that are critical in fostering bonding, trust, and emotional connection. Oxytocin, often called the bonding hormone, plays a central role in the development of attachment. It is released during physical touch, such as hugging, kissing, or sexual intimacy, and even during emotional exchanges such as sharing personal thoughts and experiences. Oxytocin has a calming and soothing effect, promoting feelings of security and comfort in the presence of a partner. It helps to solidify the emotional bond between two people, fostering trust and reinforcing the desire to stay close to one another. This shift from the high-energy, dopamine-driven phase of attraction to the calm, oxytocin-driven phase of attachment marks the transition from early love to deep, lasting love. As relationships progress, oxytocin continues to play a role in maintaining the bond. It promotes positive feelings and encourages partners to seek out connection even after the initial rush of infatuation fades. Vasopressin, another hormone involved in attachment, works in concert with oxytocin, particularly in men. While oxytocin has been more closely studied in women, vasopressin has been linked to behaviors such as protective instincts and territoriality. It is associated with the drive to commit and stay loyal to a partner, helping maintain the long-term bond that is crucial for raising children and navigating shared life experiences. Together, oxytocin and vasopressin help to stabilize the emotional connection between partners, turning the passion of infatuation into the enduring bond of love, the cognitive and emotional dimensions of love. While chemistry plays a fundamental role in love, it is far from the whole story. Love is also a deeply cognitive and emotional experience. Our beliefs, memories, and expectations all shape how we experience love and attachment. The way we think about our partner, how we interpret their actions, and how we expect them to behave influences our emotional responses, and, ultimately, our feelings of love. One of the cognitive components of love is the mental representation of the partner. The more time we spend with someone, the more they become a part of our self-concept. This psychological merging is not purely chemical. It involves cognitive processes where the brain integrates the partner into a shared sense of identity. When oxytocin is released in close, intimate moments, it reinforces the bond and solidifies this mental representation, making the partner feel more like an extension of ourselves. Additionally, the memory systems in the brain play a role in love. The hippocampus, which is responsible for memory consolidation, stores emotional memories tied to our experiences with our partner. These memories can become deeply ingrained over time, forming a reservoir of positive associations that strengthens the attachment bond. In this way, love is also about the stories we tell ourselves about our relationship, the shared experiences that we remember and reinterpret as part of our ongoing narrative, social and cultural influences on love. While biological factors provide the foundation for romantic love, social and cultural factors significantly influence how we experience and express love. Different cultures have different norms, expectations, and practices regarding relationships, marriage, and love. For example, some cultures prioritize romantic love as the foundation for marriage, while others place more emphasis on familial duty, social status, or arranged partnerships. The way we understand and experience love is shaped by cultural scripts and societal norms. The expectations of how love should unfold, whether it's the whirlwind romance that leads to marriage or a slow-building partnership, are deeply influenced by our environment. For example, modern Western society often values independence and individualism, which can shape the way love is conceptualized and expressed. In contrast, collectivist cultures may emphasize interdependence, shared responsibilities, and familial connections, altering how love is experienced in those contexts. Social media and digital technology have also changed the landscape of romantic relationships. Online dating apps, social media platforms, and digital communication tools have altered the ways people meet, connect, and form relationships. While these tools provide new opportunities for connection, they also change the dynamics of how love is formed and maintained. The ease of virtual communication, instant gratification, and the pressure to present an idealized version of oneself can all influence the experience of love in ways that might not be fully understood. The limits of chemistry, what love needs beyond biology. Ultimately, while chemistry plays a significant role in romantic love, it does not fully explain the experience. Love is not just about hormonal surges, neural activation, or chemical reinforcement. It is also about the stories we create, the values we share, and the emotional and cognitive bonds that we build with another person. Love requires ongoing communication, trust building, vulnerability, and shared experiences. It is through these social and emotional processes that love transforms from an initial spark of attraction to a deeply meaningful, long-lasting connection. For love to thrive beyond the initial rush of infatuation, individuals must continue to invest in the relationship. Emotional intimacy, shared goals, mutual respect, and understanding all play a role in sustaining love. While oxytocin may strengthen the bond and dopamine may provide motivation to pursue it, the cognitive and emotional dimensions of love help make the connection enduring. Conclusion: love is a multi-dimensional experience. So, is love just chemistry? In a sense, yes. It's grounded in neurochemicals like dopamine, oxytocin, and vasopressin, which shape our emotional experiences and attachment patterns. But to reduce love to mere chemical reactions is to miss the richness of the human experience. Love is both a biological process and a deeply emotional, cognitive, and social experience. It involves the brain's reward systems, but it also requires the heart, the mind, and the social context in which love is expressed and experienced. At its core, love is a multidimensional phenomenon. It is a dynamic interplay of chemistry, emotion, cognition, and culture. Understanding the chemistry behind love can deepen our appreciation of the powerful forces that drive connection. But it is only by embracing the full complexity of love, its psychological, emotional, and social components, that we can truly grasp its depth and significance in our lives. Belonging is a fundamental human need, deeply ingrained in our biology and psychology. The desire to be part of a group, to feel accepted and valued, is not just an abstract social concept. It is a biological imperative that has shaped the way humans have evolved over millennia. Our brains are wired for connection, and the chemistry that supports this drive is rooted in the same neurochemical systems that regulate emotions, attachment, and bonding. The chemistry of belonging is intricately linked to the release of oxytocin, dopamine, and other neurochemicals, each playing a crucial role in how we form relationships, foster a sense of community, and navigate social dynamics. The biological imperative of belonging. From an evolutionary perspective, belonging to a group has been essential for survival. Early humans lived in cooperative groups, relying on each other for food, protection, and care of offspring. Those who were part of social groups were more likely to survive and reproduce. This is why the need to belong is so deeply ingrained in human nature. Our brains are hardwired to seek out connection, because at a fundamental level, it has always been a matter of life or death. The brain areas most involved in the experience of belonging are those that regulate social behavior and emotional processing. The ventral striatum, a region associated with reward and motivation, lights up when we experience social inclusion or positive social feedback. The prefrontal cortex, involved in decision-making and social cognition, helps us assess our place in social hierarchies and navigate complex social networks. But perhaps most importantly, the limbic system, including structures like the amygdala, hippocampus, and hypothalamus, regulates emotional responses to social interactions, particularly those related to acceptance and rejection. Oxytocin, the key to social connection. At the heart of the chemistry of belonging is oxytocin, often referred to as the love hormone or bonding hormone. Oxytocin is released during moments of emotional closeness, such as hugging, touching, or sharing intimate experiences. It fosters feelings of trust, empathy, and emotional connection, making us more attuned to the people we care about and reinforcing our sense of belonging within a group. Oxytocin is particularly important in forming and maintaining close relationships, whether between romantic partners, family members, or close friends. But oxytocin's influence extends beyond just close relationships. It also plays a key role in the broader social dynamics that create a sense of belonging. When we experience social inclusion or positive recognition from a group, oxytocin is released in the brain, promoting feelings of safety, trust, and warmth. This release strengthens the emotional connection we feel to others, making the experience of being included feel rewarding. Oxytocin's role in social bonding has been well documented in both humans and animals. In prairie voles, for example, oxytocin facilitates the formation of pair bonds between mates, and the presence of oxytocin receptors in areas of the brain associated with reward and social behavior is essential for establishing these bonds. In humans, oxytocin's release during moments of physical touch or emotional connection promotes the formation of lasting bonds and encourages feelings of social safety and belonging. The more oxytocin is released, the more we feel connected to others, reinforcing the desire to stay close and form stable, supportive relationships. Dopamine and the reward of connection. While oxytocin is key to the emotional experience of belonging, dopamine plays an equally important role in the reward system. Dopamine is the brain's primary neurotransmitter associated with motivation, reward, and pleasure. It is released when we experience something rewarding, whether it's a tasty meal, a successful achievement, or social interaction. When it comes to belonging, dopamine is released when we receive positive feedback from others or experience moments of social connection. When we feel accepted or valued by a group, dopamine reinforces these feelings by signaling pleasure and reward. This neurochemical reinforcement makes the experience of social connection highly motivating, encouraging us to seek out more opportunities for interaction and group inclusion. In this way, dopamine strengthens the behaviors that lead to belonging, such as social engagement, cooperation, and emotional bonding. Dopamine's role in belonging also explains why social exclusion can feel so painful. When we are ostracized or rejected, we miss out on the dopamine-driven rewards that come with inclusion. Instead, the brain experiences a dopamine deficit, making us feel emotionally distressed and motivated to re-establish connection. The discomfort of social exclusion can feel akin to the pain of physical injury because both involve the same neural pathways. This reaction is adaptive from an evolutionary standpoint. Social exclusion was once a major threat to survival, and the brain's strong response to rejection helped to motivate individuals to seek out connection and reintegration into the group. The role of cortisol in stress and belonging. While oxytocin and dopamine are associated with positive feelings of connection, cortisol, the body's primary stress hormone, plays a crucial role in how we respond to social exclusion. Cortisol levels rise when we experience social rejection or isolation, triggering a stress response that is designed to alert us to potential danger. In early human history, being excluded from a group was a threat to survival, so the body's stress response was designed to react to such experiences with heightened alertness and urgency. Social exclusion activates the same neural circuits as physical pain, and studies have shown that increased cortisol levels are linked to feelings of emotional distress caused by rejection. The body's stress response to isolation reinforces the importance of social connection, making us feel uncomfortable and motivated to seek inclusion. This is part of the brain's natural drive to ensure that we remain part of a social network, as being excluded from a group historically reduced one's chances of survival. Interestingly, oxytocin has been shown to counteract the effects of cortisol. When we experience social inclusion or positive social interactions, oxytocin is released, which not only strengthens the bond but also helps reduce the negative effects of stress. This shows how the brain's attachment systems are intricately linked to emotional regulation and well-being. When we belong, oxytocin helps buffer the negative effects of stress and promotes feelings of safety, comfort, and trust. Loneliness and the chemistry of disconnection. While belonging has powerful positive effects on the brain, the experience of loneliness triggers a cascade of neurobiological changes that are detrimental to both mental and physical health. Loneliness is often described as a social pain, and for good reason. When we feel isolated or disconnected from others, the brain activates the same pain-related pathways that are involved in physical injury. The anterior cingulate cortex and the insular, both involved in processing social and physical pain, become highly active during feelings of loneliness, leading to emotional distress and negative health outcomes. Chronic loneliness is associated with elevated cortisol levels, which can lead to inflammation, immune system suppression, and an increased risk of cardiovascular diseases. The lack of oxytocin release in lonely individuals means that the calming, stress-regulating effects of the hormone are diminished, leaving the body vulnerable to the negative effects of sustained stress. Additionally, the absence of dopamine-driven rewards from positive social interaction reduces motivation and pleasure, contributing to feelings of emotional numbness and despair. The impact of social media on the chemistry of belonging. In the digital age, social media platforms have become a primary source of connection for many people. While these platforms provide the illusion of belonging, they often fail to provide the depth of connection that humans need to truly feel seen and valued. Social media interactions, while capable of releasing small amounts of dopamine when we receive likes, comments, or positive feedback, do not offer the same rich oxytocin-driven bonding that in-person interactions do. The superficial nature of online interactions can contribute to a cycle of comparison, loneliness, and emotional distress, particularly when individuals engage in social comparison and see curated versions of others' lives. While social media can provide a sense of belonging in certain contexts, it is important to recognize its limitations. The depth and emotional resonance of real-life connections, where oxytocin, dopamine, and other neurochemicals can work in harmony to create genuine bonds, cannot be fully replicated online. This highlights the importance of balancing digital engagement with face-to-face social interactions that allow for meaningful connection, emotional intimacy, and belonging. Conclusion: the chemistry of belonging in a connected world. The chemistry of belonging is rooted in our biology, shaped by neurochemicals such as oxytocin, dopamine, and cortisol, that regulate our social experiences and emotional responses. These chemicals reinforce the need for connection and social support, and they help us navigate the complex world of relationships and social networks. While belonging is essential for survival and well-being, the modern digital world challenges the way we experience and foster this connection. Understanding the biological basis of belonging can guide us in building healthier, more meaningful relationships and in finding ways to foster connection in an increasingly isolated world. Belonging is not just a social or emotional experience, it is a physiological one. The neurochemicals that support connection can help buffer stress, promote trust, and deepen emotional bonds. By recognizing the profound impact that belonging has on our brain and body, we can prioritize real, meaningful connections and create environments where individuals feel seen, valued, and emotionally supported. In doing so, we can create a healthier, more connected world, where belonging is not just a desire but a fundamental human need fulfilled through shared experience, trust, and love.